Hello Pasangala, welcome all of you to We Need Tamil Nadu. So most of you requested in human physiology, ma'am please do excretory product and elimination. So adinalatha in the one short video we are going to understand excretory product and elimination. So in the chapter Mudinja Appuram students, you are going to understand everything, and the clear using nice images, drawings, we are going to understand this chapter. Okay, so do not worry, just make sure that you are sitting full time in this one short video and you are not taking around 4 to 5 hours to complete this one short video. Make sure that you're sitting from the starting of the class till the ending of the class and make notes whenever possible. Scribble notes at least in your NCERT, and the notes of this will be shared on Telegram channel. We Need Tamil Nadu Telegram channel. Many of you have not joined, so please join, because later when we are giving you DPPs, PYQ practice, it will be beneficial for all of you. Okay, so start panlama students. Ore or nimisho. Okay, it's a very nice chapter. Okay, a beautiful chapter. You know, human physiology classes are always nice. So adinala don't worry about, ma'am, how it is going to be. Is it going to be difficult? No, it is not going to be difficult. In the chapter 11, the students, we can expect 3 questions. Human physiology, ella chapters mein, you can expect 3 questions from each of the chapter. Illena some chapters 2 questions. So adu vandu konjam vary aite irukum ella yearum. Last year we noticed that only 1 question came from the entire human physiology. So that way it shouldn't happen, because human physiology is very important. Like if you are a student who is going to be a future doctor, you should know human physiology. There is no skipping of it. Okay. So yes, let's start with excretion. What is this excretion? Excretion means where we are doing removal, right? We are doing bright color. Okay. We will use some other color. Adigama bright light. Okay, so excretion means, students, it is removal of waste, right? Removal of waste outside the body. That is what is happening. Yes, ma'am, that is what is happening. Now, when I say waste, do not get confused with fecal matter, okay? Outside the body. Because fecal matter is going to be removed by the digestive system, and urine will be removed through the excretory system. So here, removal of waste. Now, what is this removal of waste? What are the waste materials that is going to be formed in our body? Students, it can be HCO3, okay? It can be urea, okay? It can be chlorine, it can be salts like NaOH. Idella we can remove it from our body. Correct or not? We can remove all this from our body, and that is all going to happen with the help of this excretory system. So all these are considered as metabolic waste. All these are considered as what? Metabolic waste. Metabolic waste. Clear? So students, removal of waste outside the body. Now do not get it confused with fecal matter, because fecal matter that is done by your digestive system. Okay, that is done by your digestive system. Now students, the main nitrogenous waste that we talk about, the main nitrogenous waste in the chapter full nitrogenous waste word repeat agada. Adu nitrogenous waste na enna ma'am? And the nitrogenous waste na students, right now you have to study only 3 of them, but there are more waste actually, nitrogenous waste. So nitrogenous waste are your ammonia, okay, urea, uric acid. Okay, idita the 3 nitrogenous waste that you are going to study. In the students, the organisms which are giving out ammonia, and the organism is called as ammonotelic organism. ஒரு organism uric acid excrete பண்றாங்க. So அந்த organism க்கு uricotelic organism. Urea it is excreting out under ureotelism. Okay, so telic, telism and your ammonotelic. Okay, ammonotelic, ureotelic, uricotelic. Clear? This is what the 3 organisms that give us nitrogenous waste. இப்போ நம்ம general, okay, basic when you are studying your excretion, you always understand that students, when we say removal of waste, yes, that is one job that is done by your excretory system, that is they are doing excretion. Other than excretion, they always do one more job even without asking, that is called as osmoregulation. என்ன மேம்? ஆஸ்மோரெகுலேஷன். என்ன மேம் ఈ ஆஸ்மோரெகுலேஷன்? ஆஸ்மோரெகுலேஷன்னா ஸ்டூடெண்ட்ஸ் சால்ட் வாட்டர் பேலன்ஸ். என்னது? சால்ட் வாட்டர் பேலன்ஸ். ஓகேனா? salt water balance or else in simple words, okay, ஒரே ஒரு வேர்ட்ல அது வந்து ionic balance னு கூப்பிடலாம். okay, done. okay பசங்களா? so salt water balance or ionic balance. so normally ஒரு excretory system வந்து excretion பண்றாங்க. அது கூடாத, like with that they are also doing osmoregulation. osmoregulation na salt water balance. so இதுதான் ஒரு introduction னு சொல்லுவா students. from here on we are going to learn more about it. so first thing that you have to study is about the nitrogenous waste that I mentioned. nitrogenous waste na என்ன? ammonia இருக்கு, uric acid இருக்கு, urea இருக்கு. இப்போ the organism that is giving out ammonia, okay, we are calling it as ammonotelic organism. so first of all இது வந்து ammonotelic, ureotelic, uricotelic னு already I have written. that means what you should understand, this organism is giving out ammonia. this organism is giving out ammonia. yes ma'am, this is giving out ammonia. in the ammonia வந்து students it's a red flag. என்ன இது? இது ஒரு red flag. okay, நம்ம Gen Z languageல வந்து இது ஒரு red flag. நம்ம எதுக்கு மேம் அது என்ன ரெட் ஃபிளாக்னு? ரொம்ப டாக்ஸிக் ஸ்டூடெண்ட்ஸ், ரொம்ப டாக்ஸிக். ஓகே, ஃப்ரெண்ட் கிட்ட பேசக்கூடாது, யூ ஷுட் நாட் கோ அவுட் சைட், யூ ஷுட் நாட் டூ திஸ், யூ ஷுட் நாட் டை யோர் ஹேர் லைக் திஸ், யூ ஷுட் நாட் வாக் தட் சைட். அது மாதிரி ரொம்ப டாக்ஸிக். இந்த அமோனியா வந்து ரொம்ப டாக்ஸிக். தட் மீன்ஸ் இட் ஹேஸ் டு பி ரெடிலி ரிமூவ் ஃப்ரம் த பாடி. ஓகே, த மொமெண்ட் இட் என்டர்ஸ் இன் டு அவர் பாடி, எப்படியோ வி ஹேவ் டு பிரேக் இட் அண்ட் ரிமூவ் இட் இன் அவர் பாடி. ஸோ அமோனியா வந்து ரொம்ப டாக்ஸிக். ஓகே, ரொம்ப டாக்ஸிக். clear? so dude movie all of you have seen, I have only seen some clippings of it. so adla vandu they tell no toxic, he is so toxic, adu madri da. okay, so ammonia is the most toxic. okay, since this is most toxic enna sona, it should be readily removed. ana you don't have time, you have to remove it immediately. so readily removed. the moment it enters the body, we have to remove it immediately. now ma'am, what about, like, we talk about water, we talk about how it gets, enter the body. so students, in smaller organism, diffusion happens from the body surface. okay, inge the kidney, the function of kidney, that is when ammonia is there, we do not have time for calling the kidney and telling that you please come, you do this excretion, you do this mechanism. adukku time kediyadu. so adanale nama vandu ammonia, let's say it's in the body, it has to be diffused. it can be sometimes diffused from the body surface, not in us but in other organisms. so here there is no, let's say, lack of role of kidney, we can say. okay, lack role of kidney in removal. okay, because we do not have time, so adinale we should remove it very very fast, very very fast. so it will be like lack of kidney's role, it will be diffused or removed from the body surface. removed from body surface. புரிஞ்சுதா? okay, it's understood? yes or no? yes ma'am, this is understood. okay, this is understood. adukapram students, see, now when we talk about this nitrogenous waste, we also have to understand one point. the point is, how much water do they require to be removed, or are they soluble in water? so idu vandu readily soluble in water. okay, it will get immediately soluble in water. so here you should remember, they are readily soluble in water. readily soluble in water, that means what it is, it will get soluble in water very fast. and students, idu vandu it will break down to your amino NH3 ions. okay, not ammonium. okay, so it will get broken down. now here, as I told you, it does not have any role, kidney doesn't have role In case, let's say ammonia is in our body, okay, ammonia is in our human body, it will enter into the urea cycle. Urea cycle வந்து liverல நடக்கும். அந்த cycle தான் ornithine cycle இல்லனா urea cycle னு கூப்பிடுவாங்க. So அந்த cycleல இது automatically enter ஆயி அது it will form as it will come out as urea. Now who has this, ma'am? Who is this ammonotelic? Who is this organism? Example வந்து students, almost all the aquatic organism, because it needs water. It will be easier to for aquatic organisms to have this, no? So inga vandu example is bony fishes. I hope all of you watched the Animal Kingdom one shot. Okay, Animal Kingdom one shot is very important because there I have given you all the tricks. Bony fish, aquatic amphibians, so if you notice here, see, bony fish, aquatic amphibians, aquatic insects, idella vandu obviously they are in aquatic form. That is the reason why these people are able to excrete out ammonia. So since they are always in water, it does not matter for them like if I have water or not. But let's say if it is humans, nama vandu maximum we drink 1 liter to 2 liter water every day. Adila vandu ammonia requires so much water to be removed from the body na, it's not possible for us to afford. So adinala we are not using this. Now students, an organism that is forming ammonia, that is called as, process is called as ammonotelism. Okay, ammonotelic, this is the organism. An organism that gives out ammonia is called as ammonotelic. In the process we call it ammonotelism. Okay, in this process we call it ammonotelism. Next one, the students, urea. Okay, next one enna, urea. Ureotelic anna, this is the organism. Okay, this is the organism and the process is going to be called telism. Okay, ureotelism. Just add an ISM. Okay, now ma'am, what about this fellow? Is he a red flag? Is he a green flag? Students, he is a white flag. He is not toxic, but he is not very non-toxic also. Like, he is like very neutral. He is like, okay, I am just here. So, urea vanda compared to ammonia, less toxic irukkum. Okay, it will be less toxic compared to ammonia. Okay, compared to ammonia. Inga vanda students, kidney is going to play the role. So here, kidney will be used for removal. That means we are using kidney for removal. Now I told you, in case let's say ammonia enters into our body, okay, what will be the scenario? In case ammonia enters into our body, that ammonia will enter into the ornithine cycle. What cycle? Ornithine cycle, or it is also called as the urea cycle. Okay, it's also called as the urea cycle and it will come out as ammonia. Okay, that is how the removal happens. Ma'am, what about its solubility? How is it soluble? It is soluble in water, but compared to ammonia, it is not that soluble in water, but normally it is soluble in water. Understood so far, students? Quickly smash the like button. Okay, okay. Now students, remember, some amount of urea will remain in body. Okay, it will remain in the body. This has some job here. Okay, that is why just remember this point. Later I'll tag this point along. So some amount of urea will remain in the body. So adinala we are not completely removing the urea. Clear? Now ipa vanda students, example, which are going to be the example? Ma'am, they are not going to be aquatic for sure. They are going to be terrestrial, and since you told it is there in our body, that means first example is going to be mammals. Yes or no? First example is going to be mammals. Mammal students, terrestrial amphibians. T na terrestrial, okay? Terrestrial amphibians. So mammals are the main ones. So just imagine we give out urea. Okay, we give out urea, and some of the marine fishes also give out urea. Clear? Some of the marine fishes also give out urea. Because marine water is not the good water, right? It is fully salty. So because of them, they cannot give out ammonia. Some of them will give out urea. Next one, the students, I hope till here it is clear, super clear. Okay, let's drink some water. Okay, this. One second, let me check if the screen voice is mute. Ilana voice disturbance. Okay. So next one, the students, Uricotelic. Okay, Uricotelic. Uricotelic na ma'am, this again, this is an organism. Okay, this is the organism. That means the one that is giving out uric acid. Okay, giving out uric acid, and the process ku vanda nama vanda enna uricotelism na. Okay, label panniyachu. Next students, again, now comes the green flag. So in the entire process la, the only green flag is uric acid. Uric acid is the least toxic. Okay, least toxic. Ma'am, yedukku ma'am is the least toxic item? Why didn't mammals choose this, ma'am? Because it is least toxic, then we should have chosen it. But students, the problem is this fellow is a little expensive. Expensive, am I telling that it is expensive on any of the website? No. It is expensive means what is our currency in our body? Our currency is not 500,000, 2,000 rupees note or the money that we have in Google Pay. Nama currency vande ATP. So, idu vande konja adhigama ATP utilize pannum, idu release pandradhukku. So, adanala we cannot afford to do it. Nor we have water, nor we have energy. So, adanala we have to choose urea, students. We cannot choose a green flag until you are a green flag. Okay, so here uric acid is going to be expelled out. It requires very less water. Okay. It is, here I am writing, it requires less water to be expelled out. Okay, to be expelled out. So that is the reason, see, water is less. It is least toxic. So it should have been a good process for us. But it is an expensive process. It is an expensive process. Now you will be thinking, ma'am, as mammals, are we so poor? Yes, students, we are so poor because we studied in plants only how difficult energy formation is happening. So you can just imagine our case. Okay, it is an expensive process. That means it requires, okay, it requires more ATP. Okay, ATP, that is our money. Okay, clear? Now, example, before I tell you example, students, all of us have seen uric acid. Huh? How, ma'am? Have you seen a crow, pigeon, how it spoils your car, how it spoils your white shirt, whatever you keep for drying or, you know, to hang it in the terrace, these people will come and add their deposition. So, in the uric acid one, the majority, like as waste materials, they will be semi-solid. Okay, and this semi-solid substance, we call it as pellet. Okay, pellet. So, pellet form la irukum, semi-solid paste madri, little liquidy it will be, little paste type it will be. So, most of you would have got that blessing. Okay, please let me know in the chats how many of you have got a crow, pigeon's blessing when you're going outside. I have got on all my white clothes. Okay, every white cloth I buy, I love white. So, whenever I buy white, pigeon will be like, this is the right time for me to take revenge. So, adanala vanda it will just add all the love towards it and go. Okay, so the semi-solid paste, okay, it's also called as the pellet. Paste or pellet. This is how the substance is going to be. Now, students, example. Example, I already told you. Birds, I just now told you, pigeon, crow and all of them are going to be there. Other than that, some insects and reptiles as well. Okay, insects, reptiles, you would have seen in lizard also. Lizard waste have you seen? And some insects. Okay, this is the what we have studied so far. Clear? Clear. Yes, ma'am. Okay, you want to take a screenshot? I will just move out of the thing. Please take a screenshot of this box. It is very, very important. Very important. Okay. Next topic that we are going to go, students, is a very important topic. what is the excretory structure they have an organism? இது வந்து animal kingdomல நம்ம already complete பண்ணியாச்சு. So அதனால we have to take few topics from there and study here. So first one they have given in your NCERT is what is the excretory structure. In the excretory structure வந்து it's going to be called as the protonephridia, or in other words, which is given in your NCERT, flame cells. All of you remember flame cells? I told you flame cells is for the tough organism, and the tough organism is what? Platyhelminths. Platyhelminths, students. What are the examples that you have to remember? All of you tell me in the chat. What is the trick to remember it? The trick to remember it is tough. Okay, because it is an endoparasite, the trick to remember it is tough. Tough means T and F is going to be there. So T for Taenia solium or else tapeworm. S for fasciola. Okay, fasciola means liver fluke. Okay, liver fluke. Next one, the students, protonephridic apparum, then obviously it should be nephridia, because proto is over, then nephridia should come. So here we will have, okay, I know majority of you have not marked your attendance. Quickly mark your attendance, okay, don't be lazy. So here students, nephridia is going to be in your annelids. So here we have earthworm, okay, earthworm as the example. Okay, now protonephridia done, nephridia done. Then ma'am, what have we studied? Remember, we studied Malpighian tubules, and I told you that I will teach you this when I'm teaching you cockroach. So here we go. Malpighian tubules are present in phylum Arthropoda. So here we can write Arthropoda or most of the insects, because insects have different type of systems. So every insect need not have it. Most of the insects will have it, including your cockroach. Okay, including your cockroach. Clear? Including your cockroach. Then students, one thing that is given last time I taught you in animal kingdom was antennal glands or green glands. Remember, I wrote green glands in the notes. Green glands are actually present in crustaceans. Here they have given directly prawns, but remember, prawns is a crustacean. Okay, it will again come under arthropoda itself. So prawns. So this is what you have to remember. Don't forget, this is the main thing. Okay, main thing. I don't know why I wrote only this one in yellow color. So yes, students. Okay, and write this also in white color, so there will be a uniform. Platyhelminths. Okay, how do you remember the example? You'll remember the example as tough. Taenia or tapeworm. F for fasciola, liver fluke. Okay, so once if you see animal kingdom chapter, you will be able to remember all the tricks and write it down one more time, so you will be able to remember. Now we are going to start the main topic, human excretory system. Okay, human excretory system. Are you all ready? Okay, keep all your interest. Now we are going to actually study your excretory system. So here it is very important for you to remember. Okay, I have kidney, I have ureter, so that is very important. Okay, so start panlama students. So start panadhukku we need a beautiful pink color. Okay, so first we are going to draw our kidney. So they are bean shaped structure. So this is one kidney. We have to draw one more kidney. Yes, here we go. So we have one more kidney, and top of this kidney we have a beautiful cap like structure. Okay, which is present on the superior part, which is called as your adrenal gland. Now from the kidney, students, from the kidney, so here, now I have to draw it again. Okay. So here from the center of the kidney, there are going to be two tubules, okay, two tubules which are going to come out like this, okay, which is like this, and here also same. Here you go. Here you go. Like this, it will be extending. Now, where will it open to? It is going to open into another structure which is called as the urinary bladder. What is it going to be called? As the urinary bladder. So, in the urinary bladder, I forgot to say, students. In the urinary bladder only is going to store the urine. Clear? Okay. Wait, we'll sit and draw. Okay. This swollen part, this is the urinary bladder where the urine is going to be stored. Clear? So, this is the structure. Let's just quickly label it. Now, please remember from here you will have your renal vein. Okay. Renal vein will be there here also. Okay. Then we will also have your renal artery. Like this, renal artery also will be there. Clear? Top only should be drawn. 1 second. Students, it is all opening into the same place where the ureter is coming out. Okay. Since I already drew it, that's why I am not able to change it, make the changes. But just understand that it is all coming from the same point. Okay, same point. That is, it is all opening here. Okay, it is all opening here. I think my diagram can be little better. 1 second, students. I'll try to draw it from here only. Otherwise, you might get confused. Okay. Okay, I'll just draw like this. Okay, it's all coming from the same point from where the ureter has come out. Okay, so let's quickly label it. What is this? This is nothing but the adrenal gland which is present on the top of the kidney. Adrenal gland and everything you will be studying in chemical coordination topics, so don't worry. Then students, this is the kidney. Okay, this is the kidney, and this is what, ma'am? This is the ureter. Okay, ureter. And this is what? The urinary bladder. From the urinary bladder, students, there should be an opening, right? It should open to the external genitalia. So, there we will have the urethra. Urethra. So, this is ureter, this is urethra. Most of the time students get confused. Don't get confused. Okay, don't be confused here. Then we will have the renal. Renal means always remember it is related to kidney. Okay, renal means it's always related to kidney. And here we will have renal artery. Okay, this is your human excretory system diagram. Very easy. Yes, sir. Oh, what is this? Okay. Blink started. Okay, this is done. Understood? Now, students, we have to study certain points about kidney. Okay. Yes, certain points about kidney, like what is the length of the kidney, what is the width of the kidney, what is our human excretory system consist of. So, remember the human students, you can all join my Telegram as well for the notes. It's Gopika Biology. Okay, just search my name on Telegram. You will get my ID as well. There, all the notes are there. Okay. Human excretory system. Human excretory system has what and all? It has a pair of kidney. Yes or no? Yes, ma'am. It has a pair of ureter. A pair of ureter. Then what does it have, students? It has a urinary bladder. One urinary bladder. Please don't write a pair and come, okay, because everywhere you're writing a pair, right? So a pair of kidney is the pair of ureter is the urinary bladder is the ultimately one opening is there which is called the urethra. In this, what you have to study? You have to study the dimensions of your kidney. Kidney is how long, short, all of that. So here you should understand your kidney is going to be around 10 to 12 centimeter in length, okay, and this is going to be present in a cavity called as retroperitoneal cavity. So when you should understand that in our body many cavities are there and they will be lined also, okay. So here also it will be lined by peritoneums. We say this cavity as retroperitoneal cavity. Length is around 10 to 12 centimeter. Thickness is around 2 to 3 centimeter in thickness. And students 5 to 7 width. Okay. And it weighs around 120 to 170. Clear? So this you have to remember. So here we start. We are going to write it is around 10 to 12 centimeter in length. Okay. In length. It is around 5 to 7 centimeter in width. Quickly tell me what's the thickness. Thickness is going to be 2 to 3 centimeter in thickness. Okay, how much is it going to weigh, students? It is going to weigh around 120 to 170 grams. Okay, it is going to weigh around 120 to 170 grams. Done? Okay, easy? Right. Now, I told you that this is present towards the peritoneum, retroperitoneal. Okay. So, this is present in the retroperitoneal cavity. This you will learn more in your anatomy classes, which and all cavities, which and all organs are present. But just understand it's present in the okay. Yeah. Retroperitoneal cavity. Now this much understood, yes or no? Yes. Now I told you that this region, okay, this region where the ureter, your renal artery, everything is moving in, right? This portion, this structure, students, here, this notch kind of a structure or an opening kind of a structure is called as the hilum. What is it called as? It is called as the hilum. So hilum, you have to remember that is present towards the inner concave, okay. Hilum. One question had come in the past years, that's why I am writing it down. They will ask you which is the structure that is present in the towards the inner concave. Okay, towards the inner concave there is a notch. Inner concave. There is a notch, and what is that notch called as? Hilum, through which the ureter, artery, veins, or you can write even ureter, nerves and blood vessels also you can write. There is no problem. That is how it is given in your NCERT. If you want me to write the same thing, I can write ureter, blood vessels and nerves, okay, move inside, okay, outside and inside both. So this is what is the structure that is present. Clear? So this is the whole your human excretory system general understanding. So we have understood with the help of diagram and also writing. Now your what is that, basic NCERT lines are over. Okay, now we are going to study, students. If I take a cross section of the kidney, okay, I will try to show you that. Okay, one second, I don't have the mouse. Okay, just give me a minute. So once I will show you this. Yes. Can you see this? Okay. Yes, this is how your kidney is going to look like. Can you see? So we will try and understand which is the first part. It's called the renal artery, adrenal branch. This is the renal vein through the adrenal branch. Okay, and here you will start seeing the renal artery. You have the renal vein. Okay, this will be the renal vein ureter branch. Okay, this is the renal artery. This is the ureter again. This is the ureter. This is the kidney, right? Okay, this is the kidney. This is the adrenal gland. Can you see? This is how your kidney is going to look like. See, this is how your kidney is going to look like. And this notch which is present in the concave, this notch which is present in the concave is the one where the ureter, you can see, all the ureter, the nerves, the blood vessel, everything is entering and exiting from this notch itself. And this cap-like structure, can you see, students? This cap-like structure is called the adrenal gland. Okay, it's called the adrenal gland. Okay, this is your kidney structure for you. Once I do the cross section, I will show you more structures as well. Okay. This is the cross section actually. Now we are going to take a cross section of that kidney, and inside we can see it looks like a lot of pipe systems and everything. No, can you see arteries running, lining everywhere because blood supply has to come. Veins are also there going everywhere. And students, here you can see some funnel like structures. This funnel like structures are going to be called your medullary pyramids. Okay, medullary pyramids. And this kidney has like layers. Okay, it has covering. So remember that these people will have coverings. And here can you see the entire medullary funnel shaped structures are going to be there, and this funnel shaped structures are going to be connected. Okay, like this. Can you see it is connected to a portion, and all these portions, one second, I'll just bring it back. So look here. So here can you see all these medullary pyramids are opening into one portion. From that portion it is coming out through the ureter. Okay, it is going to come out through the ureter. Is this clear? See, it looks like a lot of pipe systems and everything, but you will understand this medullary pyramid, the urine formation will happen in the nephrons. From there all it will come through this place called renal pelvis. From there it will come out through the ureter. So I will show you this again once we complete this. Okay, once we complete the cross section, because only with my notes you will be able to understand. Okay, if I take a cross section, how it is going to look like. Clear? Okay, one second. Understood? Okay, whatever we drew right now, the kidney, we are going to draw it here once again, but a bigger kidney. So we can take a cross section. Okay, I think it has gone little. See, this is the kidney. Okay, I have to take a cross section. Just imagine it will look like a bean shaped structure, right? All of you have seen your rajma. Okay, it will take, like, if I take a cross section, I'm going to get two pieces. Now what am I going to draw? I'm going to draw what is going to happen in this one inside piece. So we will erase this. Okay, now here I told you that your kidney, first of all, understand students, your kidney has two main regions. Okay, your kidney has two main regions. This outside region where I'm putting the dots, okay, where I'm putting the dots is called as the cortex region. Okay, it's called the cortex region, just like how we studied in ovary. There is a cortex region, there is a medulla region, same like that. Wherever I have put the dots, okay, to the peripheral region, this is your cortex, and inside, right here in the center, you will have your medulla. This is going to be the medulla. This region where I am drawing the dots is going to be the medulla region. So remember that the kidney has two main regions. Okay, two main regions. Okay, now we are going to start. So in the cortex region, students, I showed you this funnel shaped structure, no? So they are going to start from the cortex and slowly they will merge into the, into the medulla. So these people are going to be funnel shaped structures like this, okay, as you saw in the diagram right now, okay, in the 3D. Here you go, like this. Funnel shaped structures they are going to be. Okay, like this, funnel shaped structures. These funnel shaped structures are called as the medullary pyramid. What are they called? They are called as the medullary pyramid. Okay, here we go. Now I told you from the medullary pyramid there are small structures which are connecting, and those structures are all going to open into one system. Can you see that? Okay, so from here students, just imagine like this, from the medullary pyramid there is structures. Okay, there from here also medullary pyramid it is there, they'll all be connected. Okay, here also it will be there. Now see, my spacing is not correct, that is why like this gaps you can see, but you can understand, no, like this connections are going to be there from the medullary pyramids, and they will all, can you see, they're all connected and they will all open into one big funnel shaped structure. Okay, they will all open into this big funnel shaped structure which is called the renal pelvis, and from there the ureter. Okay, from there, from the renal pelvis, the ureter is going to take it all the way to the urinary bladder. Can you see that? It will open into the urinary bladder. Okay. Now I told you the arteries are going to be present. Renal arteries are going to be present. Renal arteries are going to line inside also. Okay. They are going to stretch inside like this, connected like this and all that. Now I am going to not draw the full thing, but just understand the connection. Like this connections are going to be present. Same time your renal veins are also going to be there, because otherwise who is going to carry? Okay. So renal veins are going to be also present. Yes ma'am, understood. Now see, I told you that this outer layer, the outer layer of the kidney has a protection. That protected layer is called as the renal capsule, and students, this renal capsule have multiple layers. Okay, it is not just made up of one layer, it is made up of multiple layers, like one fascia layer will be there. Okay, fascia layer will be there. That first layer we call it as the fascia layer. On top of the fascia there is going to be a set of adipose tissue. Okay, there is going to be a set of, now I am drawing it just to make you understand. Please understand, the diagram will not look like this. The layers are not going to look like this. Okay. Okay, like this adipose tissue will be present, and on, okay, now let's start labeling students. It's very important for us to label and understand. Here on the medullary pyramids, I told you that the nephrons are going to be present. So remember that here, okay, we'll draw it as thin as possible. Here on the medullary pyramid, here you can see Bowman's capsule is there, PCT is there, loop of Henle is there, DCT is there, collecting duct is there. Like this, nephrons will be present. Can you see like this? Nephrons will be present. Again here I am drawing. Please remember, it is not like this, like this connection will be there. Next nephron will be opening here, that kind of connections. Okay, from one collecting duct only, many will be present. Let's say like this it goes. PCT, loop of Henle, DCT, collecting duct, like this. Nephrons will be connected to one collecting duct. Many millions of nephrons can be present. Okay, now we will start labeling. First we will label the layers. Okay, this is called as the renal capsule. First layer is called as what, students? Renal capsule. Then this layer is called the adipose. Okay, and this layer is called as the it's not proper. One second, we'll write it that side. Only that. Oh, only that. This one we will write it that side. Okay. This one we'll write it here. So first, what did I tell you? It is the renal capsule. Adakaprama, we have this which is called as the adipose. Okay. Adakaprama, this we have, students, it's called as the fascia. Okay, fascia. This full thing can be called as what? This full thing can be called as the renal envelope. It can be called as what? The renal envelope. Envelope. Have you seen envelope? For marriages and all, when we go, our parents will give money inside that. That is called as the envelope. Clear? Okay, now we will start labeling from here. So this region, okay, this region of the kidney is called as the cortex region. Okay, cortex region. Okay, and from here we will see that there are medullary pyramids. Medullary pyramids are there. Clear? Yes, ma'am. Then, students, from the medullary pyramids, there are small branches. Can you see? Those are called as calyces. What are they called? Calyces. This calyces, students, they can be minor and major. That means minor are the smaller foldings, major are the bigger foldings types. So this calyces are something like, you know, how I say, like small streets are there. No, small streets, small tube-like structures that will make and go and join to a bigger city, or small streets that will go and join to a bigger junction, like that only. In the calyces is going to collect from there. And here, can you see this funnel shape structure? Okay. This funnel shaped structure is called as the renal pelvis. Okay, renal pelvis. Now don't get confused with pelvis. Renal pelvis, don't get confused. Again, pelvis, one more you will study. So renal pelvis, this is a funnel shaped structure. Very important. Funnel shaped structure. Okay, now this renal pelvis is going to open into the ureter. This no one will have any doubts. Now students, this region is called as the medulla region, and I have to give you medulla region. Now students, one more I have to give you. From the cortex, certain structures will move towards the medulla. This is called as column of Bertini. What is it called as? Renal column or column of Bertini. There is actually structures that are moving from the cortex to the medulla. It is called as renal column or column of Bertini. Column of Bertini. Okay, regions that are moving from the cortex towards the medulla. Clear to all of you? This is understood, and this is called as nothing but your nephron. This is called as the structural and functional unit of your kidney, which is called as the nephron. So this is the cross section of kidney. Clear to all of you? Anyone has any doubts? Any doubts is there? Okay, we could have drawn one more medullary pyramid here, but it's okay. Okay, here, yeah. So this is the structure. Just look at it once. If you want to make a note, please do make a note. Done, right? Okay, now what do we know about kidney? We have to write. Before that, let me just take you here through the, so look here students, now whatever we studied, I'll show you some other diagram. Wait. Many are there, so I'm confused. Never many should be there. Many is there means Gopika Ma'am will be confused. Okay, very important class this is. I am drawing and I am also showing you 3D. So look here, can you see? This region is the cortex region, this region is the medullary region, and students here can you see the medullary pyramids are going to be present. This medullary pyramids will open, okay, with the help of small calyces, and it will open to a funnel shaped structure. This is the funnel shaped structure through which the ureter, the urine will come out. Okay, this is your adrenal gland. Can you see? Okay, this is the cross section. This is the cross section. From all the places you can see the arteries and veins are lining, because it is important. You will understand that when I teach you urine formation and the structure of tubules as well. Okay, how the veins and arteries are going to line. Okay. Wait, I'll type kidneys cross section, that time more will come. Okay, it's loading. See here students, same thing. Okay, here again you can see. Can you see there are certain regions that are extending from the cortex towards the medulla, that is called as your column of Bertini. Okay, column of Bertini, and here you can see the medullary pyramid. This white color thing that you can see is the major calyces, and you can see here minor calyces. They are all opening to a proper funnel shaped structure, which is called the renal pelvis, which is going to extend towards the ureter. Okay, and this is through which the hilum, this region is the hilum, just like how your rajma, beans and all have a small dot. Have you seen? That is like the hilum. So here also this is the hilum, and these are your medullary pyramids. Clear? I hope this is super clear for all of you. And there is artery and vein lining. Okay, artery and vein lining. Now we will go back here. Okay. Yes, this is done. Now we have to write certain things about the kidney, students, that we will write it here. First of all, what do we know about the kidney, ma'am? We know it is a reddish or a brownish, okay, reddish brown colored kidney. Okay, it is reddish brown and it is bean shaped. Yes or no? Yes, ma'am, it is bean shaped structure. How many we have? We have a pair of kidney, right? We have a pair of kidney. Now students, location, that is all very important for us. Okay. Now students, we have a pair of kidney and we know the kidney has two regions. Okay. Kidney has two regions. What are the two regions? One is called the cortex. One is called the cortex and one is called the medulla. Always remember, cortex is the outer region, medulla will be the inner region. Same thing goes for your adrenal gland, ovary, everything. Okay, this is going to be inner, the other one is going to be outer. Okay, this is the outer layer, outer region. Outer means don't think outside the kidney, you understood, no? The peripheral region. And this is the inner region. Okay. Idukapram students, we have where is the placement of the kidney. Kidney vanda between which thoracic, lumbar, where is it? Most of the people tell kidney is here. That is wrong. Kidney is here. Okay. So kidney is actually present. Kidney is present between, okay, between T12 to L3. Okay. That means thoracic vertebrae number 12 to lumbar vertebrae 3, it is going to be present. Okay, from the last thoracic to the lumbar third, it is going to be present. So that is the location. That is very, very important because this question has come multiple times. Okay, the mukyamana or topic. Done. Now I told you this renal pelvis, towards the renal pelvis, what is there? I told you there are certain projections which are called as calyces. So remember students, there are minor calyces and major calyces. Just make a note and just keep, okay? And remember that kidney's structural and functional unit is nothing but your nephron, okay? And each kidney, okay, each kidney have around 1 million nephrons. 1 million nephrons. Clear? 1 million nephrons. Done. So each kidney have 1 million nephrons, and remember the renal pelvis, how does it go to the renal pelvis? With minor, major calyces and a funnel shaped structure called as the renal pelvis. Okay. So the notes are ready for you. You don't have to write the notes. That's why I am sitting and writing everything for you people. Clear? Now we are going to the next topic, that is going to be your nephrons. Okay. Nephrons. Before we go to nephrons, students, we can write about nephrons actually. Okay. We will directly go to nephrons. We will delete this slide. Okay. Yeah. Nephrons. What is this nephron, ma'am? Nephron is basically the structural and functional unit of kidney. What is it? It is the structural and functional unit of kidney. Yes or no? Yes, ma'am. What do I mean by structural and functional unit of kidney? I would say it is more of a functional unit of the kidney because this is helping in the urine formation. This word is more appropriate because it is helping in urine formation. I wouldn't say it is structurally important, but it is functionally very important because it is helping in urine formation. Now students, nephron, when I say nephron, you should remember that this nephron has two main parts. Most of us understand nephron in a very, very wrong way. So this is important for you. Just make a note. Nephron is actually just two regions. Which are the two regions? First region is nothing but your Bowman's capsule. That means what they are trying to say, that means your nephron has two parts, okay? Inside the Bowman's capsule, what is there? Glomerulus is there. So in NCERT, instead of Bowman's capsule, they have directly written glomerulus. Glomerulus is what, students? Glomerulus is actually a tuft of capillaries. Tuft of capillaries na or bunch of capillaries, okay? Adikaprama students, we have something called as the renal tubules. Renal tubules irukku. Okay. In the glomerulus வந்து எங்க இருக்கும் students? In the glomerulus வந்து ஒரு cup shaped structure ல இருக்கும். Okay, it will be present in a cup shaped structure. So இது பார்த்தா you will understand. This is the cup shaped structure. Cup is not good. Okay. இது மாதிரி ஒரு cup shape structureல what is going to be that tuft of capillaries. Tuft of capillaries na ஒரு bunch of capillaries. இது மாதிரி highly convoluted ஒரு bunch of capillaries இருக்கும். Clear? Tuft of capillaries. So this is your glomerulus. What is going to be there, students? The one that is bringing the blood. Remember, the one that is bringing the blood. In the blood, it is bringing, no? This is called the afferent. This is called the afferent arterioles. Okay. In the blood exit agadu. So adu efferent. Exit ana E. Efferent ana exit. Adu madri. The one that is taking the blood away is called as your efferent arteriole. So E for exit, E for efferent. Okay, clear? So efferent and efferent, and in the structure of the students, it's called the Bowman's capsule. In the structure, this cup-like structure is called as the Bowman's capsule. Bowman's capsule na or cup-like structure, and the Bowman's capsule inside, what is that? Tuft of capillaries is there. Okay, so Bowman's capsule. Sometimes, students, we can also call this Bowman's capsule and the glomerulus. Okay, glomerulus as Malpighian corpuscles. Okay, what can we call this? This and this together, we can call it as the Malpighian corpuscles. All of you are enjoying? Yes or no? Malpighian corpuscles. Okay, Malpighian corpuscles. Done? Yes, ma'am, done. Clear? So glomerulus ஒரு tuft of capillaries, and the tuft of capillaries Bowman's capsule. Okay, it will be inside the Bowman's capsule, and then, students, you have one that is bringing the blood, which is called as afferent arteriole. One is that is taking the blood out, is called as efferent. E for exit, E for efferent. Okay, so this is important. Now, students, usually the nephrons. What are the parts of the nephrons? Na, we write glomerulus, we write renal tubules, and the renal tubules the collecting duct to include பண்றாங்க. But ana adu தப்பு, students, because ஒரு collecting duct la multiple nephrons can open. So we cannot say part of nephron, we can never say it is collecting duct. Okay, this question has come one of the years, that's why I'm stressing on this very clearly. Clear? So it is called as Malpighian corpuscles. All this we can also call this as Malpighian body. Okay, now remember the renal tubules have three main things. Okay, what is it? It's called the PCT. Okay, loop of Henle, and DCT. Okay, DCT. PCT, loop of Henle, DCT. PCT na proximal convoluted tubule. I should sit and write. Exercise. Proximal convoluted, okay, tubules. This is the loop of Henle. Already you know it is a U-shaped structure. DCT students, distal convoluted tubules. Okay, this is the renal tubules part. I told you collecting duct is not a part of the tubule, so never say that collecting duct is a part of the tubule, because collecting duct multiple nephrons will open into it. Clear? Done. So exciting, right? So nice to study human physiology. I want all of you to love human physiology at the end of this, one short series. Everyone should be like, yes, human physiology is so exciting, so nice it is. Yes or no? Okay. Next, we are going to study the structure of nephron. That means we are going to understand where these tubules are there, where the blood vessels are there, everything we are going to study. Clear? Yes, ma'am, clear. Okay. So let's start drawing. So students, we have our tubules. Okay, first we have to draw. Never think that this is straight. Okay, it is not. Nephrons are not going to be straight. Okay, this is your loop of Henle, U-shaped structure. Okay, and then comes your DCT, and then it goes to the collecting duct. Okay, இது மாதிரி தான் இந்த tubule வந்து complete பண்ணணும். U-shaped loop of Henle. Here we go. So students, இது வந்து collecting duct. Okay, collecting. So one nephron won't open, multiple nephrons can open. Okay, now talking about the tuft of capillaries and your blood. Okay, so English students, please understand, here you will have a tuft of capillaries. Tuft of capillaries now, a bunch of blood vessels will be there, and the blood vessels ipdi exit out. Okay, now this efferent, this is the efferent, the one that is bringing the blood. Now the efferent vanda enna பண்ணுவாங்க students, the efferent vanda, it is going to stretch towards the blood, right? Because here countercurrent mechanism has to happen. So here just remember, they're going to connect like this. Now I am drawing it very randomly because I do not want to spend a lot of time drawing this. So adinala, so in the blood vessel vanda ipdiye loop of Henle. Okay, it will extend like this to the loop of Henle, and it will, let's say, stretch up to somewhere here. Okay, this madri it will get connected. Now remember, from here the efferent arterioles, it is stretching, and it is coming to the loop of Henle, and it will be connected something like this. Okay, like this. Okay, இது மாதிரி இந்த loop of Henle வந்து இந்த blood vessel ரொம்ப முக்கியமான ஒரு this students. அதே போல தான், அதே same way இங்க வந்து ஒரு renal vein, okay, இப்படி exist ஆகும். Okay, ஒரு renal vein வந்து இப்படியே it will go out. Clear? It will go out. That also we can draw actually, but ஆனா இது ரொம்ப confusion ஆகும். Okay, it will become too confusing. That is why. Okay, now students, let's continue. So here please understand, here veins also will be there. Anna, it will be too complicated if I draw it, so adinala I am skipping it. But remember, from here also the venule connection, okay, venule connection can happen. That is, here the veins can be connected. Okay, one side la ipdi connection panna mudiyum. Okay, clear? Like this connection can come because I have connected it from here, it is not possible. Clear? Since I have connected it this way, it is not possible, but please understand, from here also veins will be there. So here you can remember that interlobular veins will be there, interlobular arteries will be there. Clear? Okay, here I will show you for arteries. Arteries, it's easier for me to show because in the artery vandi ipdi stretch pannina, I will be able to draw ipdi. Okay, yes. So this interlobular artery I can show, veins also will be there, but vein connection is not, usually we do not show because it is little complicated to show both. Okay, remember, from the renal artery it is coming, the blood is coming, so from there it will go. So this becomes your efferent arterioles. Okay, so efferent is the one, students, bringing the blood inside. Okay, it will pass on through the glomerulus and it will go down, and efferent is the one who is taking the blood. Okay, taking the blood like this and giving it to the tubules. Okay, this is how it's happening. So this is going to be called the afferent arterioles, this is going to be called the efferent arterioles, and what is this region called as? This is called the PCT, all short forms I am writing. Okay, this is called as loop of Henle. Loop of Henle, if you see, you will notice that, my god ma'am, it is fully, like, you know, full of mesh. Yes, it is like a mesh. Okay, the blood and the this one will be like a mesh. This is the DCT and this is the collecting duct. Collecting duct. Remember, from here other openings can come. Okay, other nephrons also can open here. From here the other nephrons also can open and they will just continue like that. Multiple openings will be there. Okay students, so idu vanda clear? Is the diagram clear? Now I am going to talk to you about a little more about this loop of Henle and everything. So for that I am going to remove this, okay, vein. So this loop of Henle, students, it has two regions. Enna two regions, or descending limb irkum or ascending limb irkum. Okay, so idu da descending limb. Descending loop of Henle. So this is the descending loop. இது வந்து என்ன இருக்கும்? இது வந்து ascending loop. Ascending loop. So students, please understand, in the descending loop in the ascending loop and here you can see lot of blood vessels are there. This is important for us to understand. Okay. Okay. இது வந்து students, either in the descending loop ascending loop it is permeability will change. Adu vanda I'll teach you in urine formation. Now can you see some blood vessels which are extending, full loop formation it has made in the loop of Henle? This is for counter current mechanism to happen. Okay, this is for counter current mechanism to happen. In the loop of Henle vandu or hairpin madri or structure. Okay, hairpin na enna ma'am? Now from here, let's say I'm coming to Tamil Nadu or I'm going to Kerala. So you will notice roads ku vandu ipdi turn irukum. Adha hairpin like structure. Okay, hairpin illana U shaped structure. Ipo vandu students, in the PCT la, okay, in the PCT la vandu there is some specialized cells. Okay, in the PCT, okay, in the PCT there are some specialized cells which are actually helping in increasing the absorption. Okay, this again I'll teach you in animal tissues. So in the cells, one the students, it is called as brush border cuboidal epithelium. Okay, idu madiri tha brush border na microvilli will be present on the apical surface and they will actually help in the increase in surface area. So since they will increase the surface area, it will be easy for us to do maximum reabsorption. Okay, to do maximum reabsorption. I'll write this elsewhere. Okay, this is the descending loop. Descending loop. Okay, done. So this PCT here, what is there students? It is called as brush border. Okay, brush bordered cuboidal epithelium. Okay, right now I'll just write epithelium because there's no space. Again, when I teach you urine formation, இன்னொரு வாட்டி I'll write. Okay, brush bordered epithelium. So in the PCT la வந்து cuboidal brush bordered epithelium இருக்கும். இந்த brush bordered epithelium வந்து surface area increase பண்றதுக்கு ரொம்ப அதிகமா help ஆகும். Okay na? Okay, clear? Okay, yes. So here I told you collecting that. Now I'm going to teach you how this efferent arterioles is going to split. Okay, because students, in the structure vanda, this is called as actually the peritubular capillaries. Okay, whatever I'm writing in blue, it is little important. Okay, peritubular capillaries. Okay, actually this is, here I will write peritubular capillaries because I have to do one more labeling. Don't worry, you will understand everything. Okay, don't be like, oh, I'm not understanding. Panic. Don't have panic. Straight line only is never coming. Peritubular capillaries. Okay, efferent arterioles students want the partition. Okay, divide பண்றாங்க. So it will divide into peritubular capillaries. Again, peritubular capillaries will divide into smaller blood vessels which are called as vasa recta. Okay, which are called as what? Vasa recta. Either எல்லாத்தோட use வந்து we will learn it later. Okay, we will learn it later. So what did I tell you? Efferent arterioles வந்து it is going to split. Okay, not like it is going to divide. Efferent arterioles are going to divide. What are they going to divide, students? They're first going to divide as peritubular capillaries. Ana chinna capillaries are divide பண்றாங்க. Okay, so that it can stretch through the all the tubules. Okay, peritubular capillaries. Peritubular capillaries இன்னொரு division வந்து ஒரு for smaller capillaries to happen, that is called as your vasa recta. In the vasa recta one, the students, it is only going to be present near the loop of Henle. Adanale this is important because in the vasa recta is going to help in counter current mechanism. So just remember all this. Okay, counter current. See, I am writing, I always write notes very neat, clean for you. So later you don't have to struggle making short notes. You can go through my notes and you will understand everything. Okay, so I want all of you to utilize it and also share it with your friends. Tell your friends, like this one teacher is there, she writes notes for all of us. So tell it to your friends, so let them also join. Okay, namma vande, like we should know how to help another person, right? We should know how to help another person. Clear? Okay. Peritubular capillaries. Done, done. Okay, now we are going to study type of nephrons. Okay. Should I move? You want me to take a photo? You can just take a screenshot. Okay, if you want to take a screenshot, you can take a screenshot. Okay, done. Okay, I'm assuming this is done. This also, I'll just put a box here for you to understand that this is important. Okay, yeah, this is important. Make sure that you learn all of this. Okay, next one, the student, structure of nephron. In the edathile, you can see here. Here you can see you have your afferent arterioles. So afferent arterioles is taking it from your interlobular arteries. I already told you. Now see, the afferent arterioles is entering. From there it is exiting. That will become your efferent, and it is stretching. Can you see it is going all the way fully, like, you know, convoluted it is. அது வந்து convolute. Here you have PCT. அதுக்கப்புறமா here you can see students loop of Henle. So in the venule வந்து இப்படி attach ஆகும். Okay, it will attach here in the middle, and then from there you can see it is stretching. Okay, it is stretching all the way. Loop of nephrons, that is your loop of Henle will be there. Then you will see peritubular capillaries, and then they will all stretch and they will go to the collecting duct. Okay, collecting duct. Ipo when can you see one dotted line? Adedikku students, that dotted line is to make us understand that this entire nephron is present in the cortex and this entire structure is present in the medulla. Okay, that I will teach you. That means here, let's say, okay, let's say this is the structure we have. Where is dotted lines? Students, so they are saying us that up to here, okay, up to here. Now I have drawn it a little too above, but if you notice, all this region, okay, all this region is actually present in the cortex. Okay, 1 minute. I'll extend it from here. It's not coming, students. Okay. What am I doing? So from here, this is a joke. Okay, yeah. From here till here, okay, from where the loop of Henle is extending. No, this region is going to be the cortex region. So all this is going to be present in the cortex. Okay, this is going to be called your cortex medulla junction. Okay, medulla junction, and this is going to be present in the medulla. Okay, because it will extend inside the medullary pyramid. So this region will be present inside the medulla. I hope these diagrams are clear for all of you. Yes, let's go and understand your types of nephron. Okay, types of nephron. Now students, in the type of nephron one, it is classified based on the how loop of Henle, that means the length of the loop of Henle. Okay, and the length of the loop of Henle. So remember that normally when I draw the kidney, right, when I drew the kidney, now let's just draw one structure. Okay, smaller kidneys are easier to draw. Will it come back or will it not come back? Okay, so students, this is the kidney. So in this kidney, I told you there are medullary pyramids. Yes or no? I told you there are medullary pyramids. So in the medullary pyramids, I told you that there are going to be nephrons also. Yes, ma'am, you told us. Okay. This is the medullary pyramid. Yes, this is the medullary pyramid. In the pyramid, I told you on the top of this is nephrons are going to be there. And the nephrons, when the students, it will be the cortex, like idella the cortex region la da irukum. Bowman's capsule, PCT, loop of Henle matram, it will go to the medulla, and then you are collecting that some portion will be in the cortex, some portion will be in the medulla. So in the diagram, what do we understand? We understand that some region, okay, some region of the structure, okay, some region of the structure here, let's just draw, I'll explain you, don't worry. Okay, some region, okay, some region is present in towards the medulla, right? Towards the medulla, let's assume that this is the medullary pyramid. Okay, this is the medullary pyramid, and in this medullary pyramid, okay, this is the medullary pyramid. In this medullary pyramid, this is the nephron, right? Here you have the Bowman's capsule, okay, and then you have the PCT, then you have the loop of Henle, then we have the DCT and some portion of the collecting duct, correct? So students, if you look here, you will notice that the loop of Henle, right here, this loop of Henle will stretch into the medulla, and depending on the length of the loop of Henle, I can classify my nephrons into two categories, clear? So remember, loop of Henle dips, okay, this is the word given in your NCERT, dips into the medulla, okay, dips into the medulla, done? Now, depending on this, depending on the length of my loop of Henle, depending on how much it is stretching and all of that, I have two types of nephrons. What are they? I have one which is called as the cortical nephron. What is it called? Cortical nephron. Okay. Second one is called as juxtamedullary nephron. Ma'am, so big name, ma'am. Yeah. So big name, but nothing much is there. Cortical nephron and juxtamedullary nephron. In the cortical nephron la enna nadakum students? Cortical nephron la vanda loop of Henle is not, the medulla is going to be very reduced. That means the loop of Henle is going to be short in a cortical nephron. That is LOH is short. So since this is very short, it will not stretch enough into the medulla. Correct or not? It will not stretch enough into the medulla. And since it is not stretching, you should remember that in the cortical nephron, it usually works. Okay, these nephrons are going to work in normal condition. Yenna ma'am, normal condition na. Let's take example. Okay, in the class la or student fasting pandranga. Innoru student normal condition la, they are drinking enough water, they are eating food and all. That means it's a normal condition, right? So cortical nephrons and the normal condition la work aga. வாய்ப்பு ஜாஸ்தி. ஓகே, ஜக்ஸ்டாக்ளோமுலர் நெஃப்ரான்ஸ் ஸ்ட்ரெஸ் சிச்சுவேஷன்ல ஒர்க் பண்ணதுக்கு வாய்ப்பு அதிகம். ஓகே, ஸ்ட்ரெஸ் கண்டிஷன் என்ன? நம்ம ஃபாஸ்ட் பண்ணும்போது, let's say we have not had food, we have been crying the whole day, I don't want to eat food, you will be little stubborn, no? So அந்த டைமில் இந்த juxtamedullary nephrons work ஆகும். Okay, cortical nephrons normal conditionல work ஆகும். Okay, now here students please understand, here LOH is going to be longer. Okay, LOH is going to be long. Clear? LOH is going to be long because here I told you இது normal condition la work பண்றாங்க. So this vasa recta and loop of Henle ஒரு counter current mechanism பண்ணும் students. That is basically done when our body is going through a stress condition and நம்ம urine la எல்லா water, okay, whatever water is there நம்ம reabsorb பண்ணணும். Okay, otherwise what will happen? Body, let's say our body is on a dehydration, we have not drank water, we are on fasting, so we cannot afford to lose any water. Yes or no? Let's say I didn't drink water from morning, I'm fasting. If I'm fasting, then how am I supposed to give any water in the urine form? So whatever maximum water is there in the urine, I will try to reabsorb it. My body will try to reabsorb it, and the mechanism is called as counter current mechanism. And the counter current mechanism will only happen when it is in a stress condition. So in the cortical nephrons, vasa recta theveya illaya? Please let me know in the comments. Theveya students? Ma'am, ille nanu okay ma'am. So adinale please understand, இந்த vasa recta வந்து இப்படிதான், it will be lining the loop of Henle. Okay, this will be highly reduced, it will be highly reduced. Okay, vasa recta will be highly reduced. Highly reduced, because here stress condition is not there, so vasa recta can be highly reduced. Here vasa recta is going to be present and it is not going to be reduced or anything, because this is required here. Vasa recta is present. Clear? Vasa recta is present. Done? Understood this? Okay, two types of nephron, cortical and juxtamedullary nephron. Cortical nephron where the loop of Henle is short, juxtamedullary nephron where the loop of Henle is long. In the cortical nephron la vanda students, it works during the normal situation like when we are drinking enough water and everything. Juxtamedullary vanda stress condition la, so adanaala inga counter current mechanism நடக்குதுக்கு ரொம்ப chances அதிகமா இருக்கு. So அதனால vasa recta is too important here. இங்க வந்து vasa recta will be reduced. Clear? Yes, is this clear? So depending on the loop of Henle, how much it is stretching towards the medulla, we have two types of nephron. Cortical nephron where the loop of Henle is short, that means loop of Henle will not stretch too much to the medulla. Okay, too much to the medulla. Here loop of Henle is long, that means it is stretched too much to the medulla. With that vasa recta, blood vessels are required for us to do counter current mechanism. I hope this is super duper clear to all of you. Okay, done. Can I go forward? Okay, yes. Next topic is going to be students urinary bladder. So here urinary bladder, ureter, everything we are going to study together. We will see if we can divide it. Okay, no need, we will write it here only. So students, urinary bladder structure is little important for all of you to know. Most of the time they'll write and teach you, with diagram also we can learn. So urinary bladder structure. Students, when we study animal tissues, we understand that there are two types of epithelium. Okay, there are two types of epithelium. One is called as simple epithelium, one is called as, oh, I don't like the urinary bladder. Okay. I think still it is little cross. So students, there are two types of epithelium. One is called simple epithelium, one is called compound epithelium. In the simple epithelium and compound epithelium, epithelium, there is one type of epithelium under compound, which is called as the transitional epithelium. And the transitional epithelium is also called as urothelium. So remember, under compound epithelium, okay, under compound epithelium, students, respiration and photosynthesis chapter is already done. Please வந்து playlistல check பண்ணிக்கோங்க. One shot playlist ன்னு ஒரு separate playlist இருக்கு. அந்த playlistல check பண்ணிக்கோங்க. Okay. Compound epitheliumல one category is called as transitional epithelium. And the transitional epithelium, students, it is present in the ureter, urinary bladder, in the renal pelvis. So because it is lining all these people like renal pelvis, okay, it is lining the renal pelvis, it is lining the ureter, and it is lining the urinary bladder. Ellame excretory what is that, system related. So adinale in the transitional epithelium is also called as urothelium. Transitional epithelium is also called as what? Urothelium. Clear? It is also called as urothelium. Clear? So students, this urinary bladder has this urothelium lining. That is why I am telling you about it. Otherwise, why will I tell you about it? Okay. So this epithelium is going to be lined. Compound epithelium means it will be multi-layered. So just remember. Okay. Like this multi-layered structures they will be. I am just drawing only one side, because if I draw full, it will take 2000 years later. Madri short we will have to do. So idida, okay, that means there will be one layer of this compound epithelium which will be lining. Adikaprama students, there is one set of muscles, okay, which are present, one set of muscles which are present. So just consider this as the outer border. So there is one set of muscles which are present, which are called the detrusor muscle. Actually, I should have drawn it outside, never mind. Okay, this is the detrusor muscle. In the detrusor muscle, the contraction it will help. Okay, detrusor muscle will help in the contraction. Clear to all of you? Okay, outside lining is the transitional epithelium. Okay, I will just draw this whole thing in blue so it will not be confusing. Okay, where have we drawn this? Okay, so this is the transitional epithelium. I have just drawn layers, 2 layers to show you that it is a part of the compound epithelium. So first they will have a transitional epithelium lining. After that they will have the detrusor muscle which is going to be present. Okay, in the detrusor muscle will be present. In the detrusor muscle வந்து என்ன பண்ணுவாங்க students, நம்ம urinary bladder contract பண்றாங்க. எதுக்கு contract பண்ண மேம்? இல்லைனா urine from the bladder will not expel out. So அதனால contract பண்ணதுக்கு help பண்றாங்க. Okay, and now I told you the ureter is opening into the urinary bladder. Okay, இது வந்து MSc we usually study this. Okay, ureter is opening into the urinary bladder. In the opening the students, see, it will open like this. Okay, it will open into the urinary bladder. After this opening, there is one more opening here, around here, or for the urethra, because the urine has to be expelled out. So in the structure where there is 3 opening, okay, in the triangular, inverted triangular structure, we call it as trigon of bladder. What do you call it as? Trigon of bladder. This inverted triangular structure we call it as trigon. Trigon na 3. Okay, trigon of bladder, because there are literally 3 openings to the bladder. Trigon of bladder le what and all openings are there students? In the trigon of bladder has 2 ureter opening, 2 ureter opening. Adukapurama it has 1 urethral. Okay, spelling urethral opening. புரிஞ்சதா? Okay, okay வாக்கண்ணா? So trigon of bladder வந்து 2 ureter opening, 1 urethral opening இருக்கும். Okay, அதுக்கப்புறமா students, see this you have understood, no? இந்த detrusor muscle label பண்ற detrusor muscle. In the detrusor muscle the students, it helps in contraction of the urinary bladder, because if they don't help in contraction, then how will the urine be expelled out? In the pressure kudukanume, so that contraction has to happen. The moment the contraction happen, we know that okay, it is time for us to use the restroom. So here students, please remember, here they will they have something called as the stretch receptor. Now I am just drawing it here like this, but just understand that these people have stretch receptor which are going to take the signal from the brain. Okay, in the stretch receptor is the one that is going to take the signal from the brain, and it is going to understand that okay, it is time for me to work. Okay, now students, you will be thinking, ma'am, urinary bladder, how much capacity does it have? Normally students, நம்ம வந்து usually maths class இல்லைனா hindi classல என்ன நடக்கும், நம்ம வந்து break முடிச்ச அப்புறம் that time we will not use the restroom, because we will be talking to our friends. Before the class we will want to use the restroom. So now teacher will tell what were you thinking so far, till now you didn't know, or what, that you know you have to use the restroom, adu madri சொல்லுவாங்க. So அந்த டைம்ல what is happening exactly I will tell you. So students, அந்த டைம்ல நம்ம urinary bladder வந்து let's say it is only 400 ml full. So அந்த டைம்ல maybe you would have asked the first permission. Okay, nothing will happen to you because your urinary bladder can store that much. Then the second time you would have asked when it is around 600 ml. By then you are feeling the urgency. You are feeling the urgency. Then slowly by 750 you will start doing Bharatanatyam, Mohiniyattam, all the kinds of dance that you have studied you will start doing. Maximum capacity of your urinary bladder is 800 ml. So in the 800 ml, or let's say by the time 750 ml also, it will give you multiple signals telling that it is you have to use the restroom. But Anna, let's say we will be busy, இது மாதிரி classல எல்லாம் இருக்கும். So அதனால நம்ம use பண்ண மாட்டோம். So then your internal sphincter, என்ன இருக்கும்? Internal sphincter muscle. Okay, this is important students. The internal sphincter by then will already be ready and it will open. Okay, internal sphincter. Sphincters na, these are the ones that will control the closing and opening of the urethra. Imagine there is no closing and opening, every time the urine will be expelled out. So first one is internal sphincter. In the internal sphincter one, the students, it will be involuntary. Okay, என்ன ஆயிருக்கும் involuntary ஆகிருக்கும். That means it is not in your control. It will automatically open depending on the signal that is being sent. After this, you are going to have one more, which is called as the external spinster. In the external spinster one, the students, it is going to be more like, let's say, in our control. Okay, there will be one more which is called the external spinster. In the external spinster வந்து நம்ம control இருக்கும். Okay, this is voluntary. That's why we can decide and we can control as much as possible. Now when we are traveling, okay, when we are traveling bus la they have only like one stop, so on the time la enna agum we have to control a lot, so that time we are basically controlling. Now students, the stretch receptor is very important for all of you. Okay, that means see, signal is going to come from the stretch receptor that you know it is time to release the urine. Let's say signal will come. This signal has come from where? From the CNS, from the brain it has come. Okay, from the brain this will be sent to whom? It can be sent to your detrusor muscle. This detrusor muscle will immediately start contraction and the urine will be expelled out. Okay, stretch receptor will send it to the detrusor muscle and immediately contraction will start and the urine will be expelled out. Clear? Anna students, if let's say stretch receptor is not functional, இது வந்து ஒரு question NEETல. Okay, imagine பண்ணிக்கோங்க stretch receptor work ஆகாது. Okay, a stretch receptor becomes non-functional. Okay, if stretch receptor is non-functional, if stretch receptor is non-functional, enna nadakum students, that will lead to leaking of the urine. Yes or no? Okay, because here there will be no one to control, so here leak of urine, and this happens mainly in people who are at old age. Stretch receptor on the signal it does not give properly, so adanale most of the time, that is why you will notice your grandparents or let's say anyone we know, you will notice that they will start having leaking of urine. That is because they are not able to control it. They are not able to control. Sometimes they will fit certain, like they can fit some tubes or something for let's say for people who are not able to, like urinary bladder becomes weak, all these muscles will become weak. Once the muscles become weak, then the signals will not be sent properly, so that time continuance will happen. That means the leaking of urine will happen. Clear? Okay, this mainly happens students in people who are old age. Okay, or old age or some disease should be there. Yes, this is done, I hope. Okay, notes are done. Okay, yes. Okay, next is going to be juxtaglomerular apparatus. Juxtaglomerular. Juxtaglomerular, no, juxtamedullary. Okay. Okay, students. So, next one, the juxtamedullary apparatus. In the apparatus one, the students, it actually works when the NaCl level or the filtrate has some problem. GFR, the glomerular filtrate has some problem. All this you will understand in the later when I am teaching you mechanism. Right now, we will just try to understand this entire process. Okay. So, again, this is your Bowman's capsule. Okay, this is your Bowman's capsule. It will go like this. Now, what is going to happen? This structure, here you will see PCT. Yes, ma'am, here I will see PCT. Okay, and then students, you will have loop of Henle, and after the loop of Henle, you are going to have PCT. Okay, wait. Like this, two structures I am drawing. Then you will have loop of Henle, and then you will have DCT, and then you will continue like this, right? So, this is how the structure is going to be there. Now, the DCT, I have to, students, understand this DCT, no, it will come actually very closer to the glomerulus. Okay, like this loop of Henle will be there. It will be very highly convoluted. So, it will come very closer to the glomerulus. We cannot draw that much because அது வந்து கொஞ்சம் அதிகமா constrain ஆகும் and you will not understand. But understand that these people will be very close, and what these people are going to help, they are going to help in understanding the filtrate concentration. So, this is your juxtaglomerular apparatus. Don't worry, you will understand. So, what is this juxtaglomerular apparatus? I told you it works on emergency situations where the NaCl count is very less, low GFR. That time, these people will work. So, now, ma'am, what is this, ma'am? What is this apparatus talking about? First of all, let me make you understand. In the DCT, okay, in the DCT level, the students, there is going to be a set of special cells, okay, which I am going to draw here. Okay, set of special cells are going to be there, which are called as your macula densa cells. Okay, macula densa cells. From there, we are going to draw it here. Okay. So, this is how your DCT, I have taken a cross section in the DCT. In the DCT, inside the DCT students, there will be certain number of cells. Okay. There will be certain cells which will be arranged like this. In the cells, we call it as the sensory cell, Elena, macula densa cell. Ma'am, what is it used for? What is its purpose? So, students, in the cells, it will identify if the GFR has some problem. Mainly, they will identify if the NaCl has some problem or not. What is the cells called as? They are called as the macula densa cells. Okay, macula densa cells. In the macula densa cells can also be called as sensory cells. What will they do? They will be able to identify. Okay, they will be able to identify low GFR, okay, and a high GFR. They will be able to do what? They will be able to identify low GFR and high GFR. That means what? Mainly they will be able to understand the NaCl concentration. That is based on NaCl increase aguma, decrease aguma. Based on that, they will be able to know this. Okay. Now, students, inge vanda, ma'am, once this macula densa cells get to know, will they do something? Yes, students, they will do something. What they are going to do? They are going to alert, that is, they are going to tell the afferent arterioles. This afferent arterioles have the smooth cells, okay, here. The smooth, some smooth cells of the afferent arteriole will get modular. They will modify themselves, okay. Some smooth cells on the afferent arterioles will do, they will change to what? Sorry. Okay. Some smooth cells of afferent arterioles modify students, they will become granular cells. Okay, in some diagrams you will see granular cells. Do not confuse yourself. In the granular cells is also called as juxtamedullary juxtaglomerular cells. What a minute, students. Okay. Okay, this is not medullary. I wrote it wrong. This is called as juxtaglomerular. Because JGAL. Juxtaglomerular apparatus. So, in the cells it will be called as juxtaglomerular cells. Also called as juxtaglomerular cells. In the cells, the students, they are granular. That means they can store or secrete certain enzymes. So, these people will store. They can store and release renin enzymes. Release what? Renin enzyme. Okay, they can release renin enzyme. In the renin enzyme, the students, it will do certain job where it can fix the GFR. Because in the macula densa, they understood that there is some problem, right? There is some problem with the glomerular filtrate rate, right? GFR has some problem. So, in the GFR fix pannadekka, we need renin enzyme who is going to do the job. So, listen to me carefully what is happening. The filtrate will be passing on, okay, the filtrate will be passing on from the tubules. As it reaches the DCT has macula densa cells or sensory cells, and the cells it will understand that if the NaCl is high or less, that means if the GFR is low or high. What it will do, this macula densa cell will send signal to the smooth cells of the afferent arterioles telling that you modify, you start releasing renin. So they will get modified to juxtaglomerular cells, or else it is also called as the JG cells. Okay, the JG cells, or else it's also called as the glandular cells. And this JG cells will store and release renin, and the renin one, it will continue for RAS mechanism. So when I teach you the mechanism, you will understand. Okay, when I teach you the mechanism, you will understand. Don't worry, everything will make sense when I move forward in the chapter. When I teach you urine formation, you will understand what is this glomerular filtrate rate. Like that, every topic you will understand. Clear? So just understand that from the glomerulus, okay, from the glomerulus, the filtrate is moving inside. In the filtrate, when the PCT in a pass I, it will go to loop of Henle, and when it reaches DCT only, it will understand that there is some issue. There is some issue. Understood? So that is the filtrate. So when the filtrate has a problem, everyone will understand, okay, it is time for me to get activated. So idu vandu continue agum students in RAS mechanism. RAS mechanism, your renin's function you will understand. But just know that macula densa is only a sensory. It can only sense if NaCl is more or high. But the function, the job will be taken care by renin. And renin will continue in the RAS mechanism. So I hope this is clear to all of you. Juxtaglomerular apparatus, or else it is also called as JGA apparatus. Clear? Clear? It's clear. Next we will start urine formation. Okay? Next we will start urine formation. That's why. So I want all of you to understand this. Otherwise, you will not understand RAS mechanism. You will not understand RAS mechanism. Understood, ma'am. Very nice. Energy is there. Quickly smash the like button also. Yes, students. Let's continue. Okay. Next is very important topic, urine formation. Okay, urine formation one the very important topic. In the urine formation, students, mainly 3 steps are going to happen. One is called as ultrafiltration. Okay, one is called as ultrafiltration. One is called as reabsorption. Okay, tubular reabsorption. One is called as tubular secretion. Okay, tubular secretion. Understood? So 3 main steps are happening. One is called as ultrafiltration or glomerular filtration, tubular reabsorption, and tubular secretion. So whenever we draw the glomerulus, or whenever we try to understand it, you should understand that this is happening in the glomerulus. Yes or no? Yes, ma'am. This is happening in the glomerulus. And these 2 will be, since the word tubular is given, these 2 words will be in the renal tubules. Understood? These 2 words will be in the renal, that is, these 2 processes will be happening in the renal tubule. Now, one line of NCERT they have given, how much blood is getting actually filtered out, because நம்ம வந்து excretion பண்ணும்போது blood in we are taking the urea, we have to throw it out. So how much blood is getting purified? Please understand, around 1000 to 1200 ml, okay, is getting filtered out in the kidney, okay, of blood is getting filtered in the kidney. That is almost how much, students? Our total, we know, right? We have calculated in body fluids and circulation, it is around 5000. So here we know that it is this is equal to 1/5 of the total blood. Yes or no? Of the total blood? Of the total blood. Yes, of the total blood. So just remember this, 1000 to 1200 ml is getting filtered out. That means, students, please remember, whenever the blood is entering into this, we call it a filtrate. Okay, we call it what? A filtrate. So a filtrate per day, how much filtrate is going to come out? Per minute, around 125 ml is going to come out. Okay, and in a day, 180 liter is going to come out. 180 liter is going to come out. In this, only 1 to 1.5 liters of urine is only going to be expelled out. This indicates what? Ma'am, this indicates that 99% reabsorption is happening. Okay, only 1% is going out, right? So students, out of 180 liters, just 1 to 1.5 liters of urine is expelled out. Understood? Yes, this you have to remember. This will come as a question. Okay, this I hope it is very clear. Now students, one more calculation that you have to know. You know, no stroke volume. Okay, stroke volume is around 70 ml, right? How did we, this I already, like in body fluids I'll be teaching you. So is around 70. Okay, now when we have to calculate the total heart blood pumped through the heart, that will be called as the cardiac output. Okay, what will that be called as? Cardiac output. And the cardiac output, students, adukku oru formula irukku. Like how do I calculate the cardiac output, ma'am? And the cardiac output calculate panadukku, you have to calculate stroke volume into the heartbeat. Okay, heartbeat. So stroke volume is around 70, heartbeat is around 72 times, right? So that is equal to 5040 ml, which can be approximately written as 5 ml. So adinale cardiac output vandu totally 5 ml irukum. Adile 1000 to 1200 ml vandu kidney na filter out agum. Okay na? Okay. Now we are going to study, students, urine formation. We are going to study first one, glomerular filtration. Okay, we are going to study what? Glomerular filtration. Romba easy. Okay, romba easy. Don't worry at all. Glomerular filtration means what, ma'am? It is already happening in the glomerulus, right? But that is not the important part here. In glomerular filtration, students, nama glomerulus, it has three main parts. Glomerular filtration happens with three main parts. Okay, just like respiratory membrane we studied in breathing and exchange, we had endothelium, epithelium. Adu madri da ingeyum and the blood vandu when it is passing, it is going through three layers. Enna ma'am, and the three layers, students, first layer is going to be called the endothelium. The endothelium of glomerulus. Okay, endothelium of glomerulus. Innonu vanda, it is called the basement membrane. In the basement membrane vanda non-cellular layer irukum, students. That means this is not made up of cell, it is just a cells which are present in the like space. Okay, adukapram, students, you will have epithelium. So endothelium irukku, or epithelium is there. Okay, epithelium of, here what will be the epithelium? Can you think it? Tell me. Epithelium of who will be there? Bowman's capsule will be there. Okay, epithelium of the Bowman's capsule. Understood this? So endothelium of the glomerulus, basement membrane, epithelium of the Bowman's capsule. So what does it do, students? It filters out the blood. Okay, it filters out the blood without taking in the larger proteins. That you have to remember. Okay, what is happening? It filters out everything, but it does not filter out the large proteins. Adanale in the glomerular filtration can also be called as ultrafiltration, because almost everything is getting filtered. Okay, almost everything is getting filtered except for large proteins. Okay, it filters everything except large proteins. Okay, புரிஞ்சுதா students? Okay, so except large proteins almost எல்லாம் வந்து filter பண்றாங்க. So அதனால அது வந்து ultra filtration னு கூப்பிடுவாங்க. Glomerular filtration also called as ultra filtration. Okay, now we are going to understand how is this layer formation, that is how are these layers formed. So first one, the students, நம்ம வந்து endothelium of the glomerulus draw பண்ணலாம். See, I am not drawing the your what is that, NCERT, there is no diagram, but just understand the layers. Okay, just understand the layers. Idukapram, students, there is going to be one layer which is the basement membrane, and the basement membrane vandu non cellular agirukum. Ana also let's just draw it. Okay, basement membrane will be there. In the basement membrane, okay, idu madri irukum. Adukapurama, students, comes the most important part, that is the epithelium of Bowman's capsule. In the epithelium of Bowman's capsule, students, one that it has a very like different type of cells which are arranged in an intricate manner, and the cells we call it as podocyte. What am I trying to say? I am trying to say the filtration is happening in such a way that we have, students, ipo example vandu chai. Okay, tea பண்ணும்போது enna nadakudhu, we have a sieve. இல்ல அதுக்கு என்ன சொல்லுவாங்க, சன்னியா சீவை, அந்த சீவை இல்லைன்னா அந்த ஃபில்ட்ரேஷன் ஃபில்டர் கிடையாதா, அதில் என்ன ஆகும், அந்த டீ லீவ்ஸ் இல்லைன்னா பிக் கிரானுல்ஸ் ஆஃப் டீ பவுடர் சுகர் எல்லாம் வந்து அங்கே it will get settled, only the tea will come out. அது மாதிரி தான் இந்த blood when it is getting filtered out, we have to remember that the filtration is happening in such a way that only the everything is getting filtered out except for large proteins. So can you see, from all the spaces the filtration is going to happen and it is going to pass on. Okay, it is going to pass on and it will enter into the PCT. So this is how filtration is going to happen. So it happens with the help of 3 layers: endothelium, basement membrane and epithelium. Now remember, this epithelium, that is the Bowman's capsule, have a special type of cells which are called the podocytes. In the podocytes வந்து students ஒரு intricate manner la arrange ஆகும். Okay, it will get arranged in a intricate, okay, it will get arranged in an intricate manner, and the intricate, because of the intricate manner, they are going to have certain spaces. Okay, இது மாதிரி certain spaces between them which are called as the filtration, which are called as the filtration slits இல்லைனா slit pore, slit pore. புரிஞ்சதா? Okay, it's called as filtration slit இல்லைனா slit pore. Clear? So they are arranged in a certain intricate manner, because of which there are going to be spaces in between. Those spaces are called as filtration slit or the slit pores. Clear? So this is the 3 layers. Okay, now when I draw it here, okay, when I draw it in the Bowman's capsule, okay, like this it will be. Now remember students, the glomerulus is going to be there. Okay, okay, like this the glomerulus is going to be there. So on the glomerulus one, the students, this first layer is going to be there, that is the endothelium. Like this endothelial cells are going to be present. Okay, through the endothelium it is going to come to the basement membrane, and the space is the basement membrane. Adukapprama here on the Bowman's capsule students, there are certain type of cells. Okay, idu vandu erase panni elro, I have to draw that again. I'm drawing all diagrams so it becomes easier, because NCERT la diagrams are not there, so most of the students do not understand it. Okay, students, idu vandu you have here, I'm drawing with the same color. So these podocyte cells which are arranged like there are, okay, like this, and remember podocyte cells will be there present like this, and between that what you are seeing is the filtration slit. Okay, so what will happen students, in the blood the blood will start coming. Okay, vanda ipdiye it will start moving, ipdi it will go. Okay, and it will go to the PCT. Clear? Yes or no? So this is the structure. I hope it is clear, super duper clear to all of you. So please understand students, this two, okay, these two are going to be cellular layer. Okay, இது வந்து non cellular layer, இது வந்து cellular layer. Okay va, வந்து cellular layer. Done? Yes ma'am, done. Now you are going to understand urine formation. Adukku munadi students, I have put a video yesterday how to improve in your mock test, and also respiration one shot, reproductive health one shot only for girls. So in the videos ella check பண்ணிக்கோங்க, especially mock test strategy, அது ரொம்ப important, because இப்போ வந்து நீங்க one shot video ரொம்ப preference கொட்டி you will not be able to give preference to mock test. So அதனால that's why you should know now itself how to improve your mock test, so it becomes super easier for you in the future, that okay, I have to study this, I have to know this is how I will improve my scores. Clear? Okay, now we are going to understand urine formation eppadi nadakumnu. So students, here you can see I have put the NCERT lines. Proximal convoluted tubule, PCT, is lined by simple cuboidal brush bordered epithelium. This I already explained to you before, which increases what? Which increases the surface area for reabsorption. Nearly, here very important line, okay, nearly all of the essential nutrients, okay, and 70 to 80% of electrolytes and water are reabsorbed in this segment. Okay, now PCT also helps in what, students? It also helps to maintain the pH and the ionic balance. pH and ionic balance on the salt and water balance, okay, and the ionic balance also it will maintain fluids by selective secretion, selectively secreting it, okay, selective secretion of what? Hydrogen ions, okay, ammonia into the filtrate and by absorption of HCO3, okay, and absorption of HCO3 from it. Clear to all of you? So PCT does the 70 to 80% of reabsorption. Adanale adukku vandu cuboidal epithelial cells with brush border epithelium we have given, and the foldings vandu it increases the surface area, and when it increases the surface area, reabsorption becomes much easier. Clear? So adu madri tha in the foldings will help in maximum reabsorption. So 70 to 80% can get reabsorbed. Clear to all of you? Yes or no? Now I'll teach you one thing to, oh, I don't want this, wait, this will become grey color, students. We'll take it from down. Okay, now when you understand about reabsorption and secretion, I want you to understand one thing, because most of the students don't understand this. Okay, students, you should tell me in the chat box how many times I drew the nephron today. Okay, that is your task. Let me see how many of you will have the concentration span. Okay, very simple I'm drawing, because here the goal is to explain and make you understand. The diagram is just for that. Okay, so in the tubule, in the tubular one, the students, reabsorption and secretion will happen. Now in your NCERT they have drawn two arrow marks. Okay, two arrow marks. First arrow mark is going to indicate, okay, first arrow mark is indicating first arrow mark is indicating reabsorption. Reabsorption na ena, let's say something is needed for my body, under the my body will start reabsorbing. That means my body wants it, so it will take it from the body. So inga vandu tubules illa. So here on the tubules above there are blood vessels. So the blood vessels will start doing reabsorption. Clear to all of you? So the arrow mark indicating like this shows that the body is reabsorbing. Okay, the body is reabsorbing whatever we want. Okay, whatever is required for the body is getting reabsorbed here. Now when I show arrow mark this way, okay, when I show arrow marks towards the tubules, understand that my body is secreting whatever it does not want into the cells. Okay, into the cells. Okay, into the cells. So this is what is happening. Yes, ma'am, this is what is happening. That means, students, here this arrow indicates reabsorption. That means whatever the body needs, okay, whatever the body needs, it will reabsorb from the tubules, telling that I want this. Okay, and whatever the body does not want, it feels like, oh no, I don't want it, it will do secretion with the help of this. Okay, it will do secretion with the help of this. So please understand this arrow mark, otherwise it will become difficult. Okay, now to remember what and all is getting secreted, that is the body does not want, remember NH, like NH4, no, like that, remember NHK. NHK na, N for, students, it is going to be ammonia. We know that ammonia is something that we cannot tolerate at all in our body. H for what it will be? H plus ions. Okay, H plus ions, and K for K plus ions. These three we are getting secreted. That means we don't want, you take it. Okay, we are telling. Then, students, what we will do? Reabsorption we will do. We will take the salts. Okay, here NaCl, NaCl. All this will be used for us later. Okay, used for us later. So remember that. Now, one more thing I want you to remember. There is two types of, you know, reabsorption to happen, active and passive. That means if let's say I want to reabsorb something, I have to utilize energy. Okay, so that to remember that, you remember gang. Okay, I think this is some gaming word. For G for glucose. That means, students, if I want to reabsorb this, I need to utilize energy. I have to take the energy. If I don't give them energy, they'll be like, no, I'm not going to work for you. Okay, I will not get reabsorbed. So whenever I want to reabsorb these people, amino acids, glucose, okay, these are, so remember like this, gang, the gang people are very active. Gang means what? They're very active, no, like that only, gang. Okay, gang. Clear? N for sodium, Na plus, and K for K plus. So idana reabsorb pannadukku they have to be active. Okay, they are active. That means they require energy. Passive means, students, nitrogenous waste. If I want to reabsorb nitrogenous waste, if I want to reabsorb water, if I want to reabsorb HCO3, all the time I can utilize these people. Clear? Yes. Yes. Okay. Done. Understood? Okay. Okay. This is the reabsorption and secretion. Now we are going to learn about the next tubule, that is the loop of Henle. So loop of Henle, students, reabsorption is minimum. Okay, reabsorption is minimum in loop of Henle. But in its ascending limb, however, this region plays a significant role in a maintaining of high osmolarity of medullary interstitial fluid, which you will understand when I teach you counter current mechanism. Okay, they do what? They will make sure that the medullary interstitial fluid osmolarity is maintained. The descending limb, okay, the descending limb of loop of Henle is permeable to water. What are they saying? D is permeable to water. D likes water. Okay, descending loop likes water and almost impermeable to electrolytes. This concentrates the filtrate. So the one who is going to concentrate the urine or the filtrate is going to be loop of Henle. Okay, one of the PYQs it is. The ascending, okay, the ascending limb is impermeable. Ascending limb, one the students, it does not like water. So A hates water. Ascending limb is impermeable to water, but it allows the transport of electrolytes actively or passively. It will allow the movement of electrolytes with energy or without energy. Therefore, as the concentration filtrates pass upwards, it gets diluted. Why? Due to the passage of electrons to the medullary fluid. This you will understand when I teach you counter current mechanism. Okay. Now, students, this one, distal convoluted tubule. Distal convoluted tubule, conditional reabsorption. Conditional reabsorption of Na plus and water takes place. Normal nadakada, but when let's say I want an emergency situation, conditional reabsorption, this people will do. DCD is also capable of reabsorption of what? What is it capable of? Reabsorption of HCO3 and selective secretion of hydrogen and potassium ions. Selective secretion of hydrogen, H plus and potassium ions, and NH3 to maintain the pH. I told you, NHK. Okay, NHK selective secretion will happen. So, secretion NHK, reabsorption gang. Okay, and why do they do this? Because they have to maintain. They have to maintain the pH and the sodium potassium balance. Okay, they have to maintain the pH and the sodium potassium balance. Clear? Understood? Now here you can see, students. Okay, here you can see. Here this is the reabsorption. In the K one the inga varanam. Okay, K plus you write it here. So here you can see reabsorption. What and all we are reabsorbing? HCO3 we will reabsorb, NaCl we will reabsorb, water we will reabsorb, nutrients we will reabsorb. What is the body giving to the tubules? H plus, NHK, NH3. Why? To maintain the pH and to maintain the osmolarity. At the same time to maintain the sodium potassium pump. Okay, to maintain the sodium potassium pump. Okay, to maintain the sodium potassium pump. And then we have descending loop of Henle, ascending loop of Henle. So in the ascending loop la vanda electrolytes movement will happen aram se, because adu vanda it is only impermeable to water. Inge vanda students, water will get continuously released. Okay, it is permeable to water, so water will keep going out. Here electrolyte will keep moving. Then we have thick segments of ascending limb. And then we have there also you can see NaCl, H2O, HCO3, K plus and H plus. Okay. Yes. Clear? Collecting duct. This long duct extends from the cortex. Okay. Extends from the cortex of the kidney to the inner parts of the medulla. This we already understood, right? This one the larger amounts of water could be reabsorbed from this region to produce a concentrated urine. Okay. So we can do reabsorption from this region to concentrate the urine. Okay, to concentrate the urine. This segments allows passage of small amount of urea into the medullary interstitium. So what does it do? It allows the passage of small amount of urea. Idella vanda You have to remember because later I'll ask you all these questions. Amount of urea into the medullary interstitial fluid. So students, between the blood vessel and the tubule, the space is there. No, this space is called as medullary interstitium. And the medullary interstitium la concentration increase பண்ணதுக்கு நாம வந்து urea, NaCl எல்லாம் யூஸ் பண்ணி நாம osmolarity maintain பண்ணணும். Okay. To keep up the osmolarity, it also plays a role in maintenance of pH and ionic balance. Okay. And ionic balance of blood by selective secretion of H plus and K plus. Clear? So you should know from where what is getting secreted. At the same time, you should also know who are the ones who are maintaining the pH, who are the ones who are helping in the other process. So இப்போ வந்து students, we are going to do counter current mechanism. After we do counter current mechanism, we have to learn all the mechanisms. Okay, we have to learn all the mechanisms. Take a 2 minutes break. Done? 2 minutes is over. Quickly subscribe to the channel. We are going to become, we are not near to 10k, but we'll become 10k. Okay. I hope all of you are checking all the videos. Yella video mein romba useful ana videos. So don't be like, should I watch or not? So students, start panlama counter current mechanism. In the ma'am, what is this counter current mechanism? Nama one the students, I told you, in stress situation, let's say in stress situation, I want to reabsorb water from my body. For that, I have to build a system who can reabsorb the water, because normally when the PCT is doing reabsorption, yes ma'am, loop of Henle is doing concentration of the urine. How is it doing? With the help of counter current mechanism. And DCT also is helping in reabsorption, conditional reabsorption. Collecting duct also is helping. Ana namma stress situation la vanda namma extra water we have to reabsorb. So adanale we have one mechanism called as counter current mechanism. In the mechanism on the students, mainly it is to maintain the osmolarity. Okay, to maintain what? The osmolarity. So in the osmolarity maintain pannadakada, in the counter current mechanism is present. So please understand, this is the loop of Henle. Okay, this is the loop of Henle, and around this loop of Henle there is blood vessels. Remember I drew this? Okay, there is blood vessels, right? This same thing I am going to draw it here magnified. Okay, this small portion I am going to draw it magnified. Now please remember, this red color what I have drawn is the vasa recta. Yes or no? It is the vasa recta. So I am writing VR. In the vasa recta, students, what is this vasa recta? This is the ascending vasa recta. Okay, so I am writing AVR. This is the descending vasa recta. Tube in loop of Henle, what are they? This one is called as descending loop of Henle, and this is ascending loop of Henle. Yes or no? So first counter current mechanism is going to happen between these two, and then those two, and then between these two and these two. So totally 3 counter current mechanisms are there. Counter current in students, this whole counter current means when two tubules who are at close proximity, in them 2 different fluids are flowing in opposite direction. So let's say filtrate is going downwards, blood will be going upwards. So that means what? In 2 tubules who are in close proximity, when the fluids are moving in opposite direction, that is called as counter current mechanism. Counter and students, nama vanda YouTube illana friends kita and all we tell, no, oh nalla counter solra. That means what? Whatever I am telling, he is giving me back answer. Adu madri tha. So idu adudha counter current, opposite. That means I am here, ascending vasa recta, there is descending vasa recta. Next to ascending vasa recta, there will be descending loop of Henle. So this two will become in countercurrent because they are in opposite, right? They are opposite and the fluids are also flowing in opposite direction. So let's draw this, okay? So here we have loop of Henle. Okay, this is loop of Henle. Okay, then students we have vasa recta. Okay, we have vasa recta. Correct or not? Yes. Now, what is this medullary interstitium, ma'am? Everyone are talking about it. Students, this is the medullary interstitium. This space that you are seeing here, no? This is the medullary interstitium. Clear? That is the medullary interstitium. In the medullary interstitium, we have to increase the concentration. Why? Because if I make this solution, just imagine, very simple, okay? If I make this solution hypertonic, that means I make it rich in electrolyte and less in water. Don't you think water from the tubules will start coming out? Yes, ma'am, it will come out. So, can I increase the urine concentration? Yes, ma'am, you can increase the urine concentration. So, normally, urine flow agumbodha, I cannot increase it. But, if I create a hypertonic solution, electrolytes increase pani, water kami pani na, I can definitely make the water to come out. Correct? Yes, ma'am, you can do that. So, here to do that, I have to go through a process. I have to go through a certain set of processes. That is what you are going to understand. See, students, you already know descending D is in love with W. Okay, D loves W. That means D is permeable to water and not permeable to electrolyte. Okay, that is descending loop of Henle. Ascending loop of Henle is in love with electrolyte. It has permeability to electrolyte, but it is not permeable to water. So, from here, you already know that the water molecules will come out. Right, water molecules will come out, but that's not enough for us. We want more water molecules to come out because it's a stress situation. Okay, so let me just write here. AVR. So, can you see counter? One is descending, one is ascending. Okay. Now, what is happening, students? Please understand. The filtrate is coming. Okay. In the filtrate, I can normally remove the water, but it is not enough. When the filtrate reaches the ascending loop of Henle, in the ascending loop of Henle, what will happen? The NaCl will start coming out. Yes, ma'am, NaCl will come out. Okay. I think. Okay. From here, what will happen, students? NaCl will come out. Yes, ma'am, because ascending loop of Henle is permeable. Here also NaCl will come out. Now, this NaCl will be picked up by who? The descending vasa recta, and this descending vasa recta will carry it all the way and it will start adding it here. Okay, it will start adding it here and it will increase the concentration of this liquid. Okay, it will try to make it more concentrated. Okay, it will try to make it more concentrated. So, let's put dots for NaCl. Okay, so now NaCl has become highly more in the medullary interstitium. So, that means the salty solution it has become. Solution has become hypertonic. Okay, solution has become hypertonic in the medullary interstitium. Now, students, it is not enough. I want more concentration. So, adinale what I am going to do, from the loop of Henle, DCT will come and after that collecting duct will be there. No, this collecting duct will keep some urea. Remember, in the starting of the class I told you, not all urea will be removed out. Some amount of urea is required in the body. So, and the urea from the collecting duct will also start moving towards this medullary interstitium and this will start increasing the urea concentration. If student solution on the hypertonica, yes ma'am, solution is hypertonic. So, students, please remember, here we do not use the word hypertonic, hypotonic. Here we use the word the osmolarity. So, osmolarity one, the students, from 300 it has reached up to 1200. Okay. Osmolarity has increased. So, here automatically what will happen, students? The water from the tubules will start coming out. This will lead to highly concentrated urine. Yes or no? water molecules will start continuously coming out, because here it is rich in electrolyte. So because of that, the urine that is going to be passed out is going to be rich, I mean it will be very, very concentrated. So by the time urine reach here, it will be concentrated. In normal situation, it won't be like that. This is only in stress situation, okay? Urine will be concentrated. So now students, remember, osmolarity has gone from 300 osmol to 1200 osmol. Okay, this question has come, so please keep in mind. Please keep in mind. Okay, now here, this is clear. Counter current mechanism also is clear. Yes, so this is the medullary interstitium. We are basically increasing the concentration in the medullary interstitium. How? By taking the NaCl from the interstitium. Yeah. We are taking the NaCl from the ascending loop of Henle, passing on to the vasa recta, and vasa recta will keep adding it here. And because the NaCl and the urea that is coming from the collecting duct is present, this urea and NaCl together will make the solution very hypertonic, increasing the entire concentration of the medullary interstitium. Automatically what will happen, students? The water will start coming out, and this will lead to the urine being extremely concentrated. Clear? Yes. So here I was telling that the osmolarity is going to go from 300 to 1200. In the entire process, basically what are we trying to do, students? We are basically trying to make sure that we can increase the urine concentration. So in stress situation, all the kind of fluids that we need to reabsorb, all that we can reabsorb into our body. Clear? That is why we are doing this process. This is called as counter current mechanism. Please remember, counter current mechanism, here 3 counter currents are there. That is, one is between, students, this is the loop of Henle. Yes ma'am, this is the loop of Henle. So between these two, that is descending loop of Henle and ascending loop of Henle, or counter current mechanism irukku, because idu rendilume direction opposite da irukku, right? Because in here fluid is flowing like this, here fluid is flowing above. Okay, then inga vanda vasa recta, students, vasa recta also, what is happening? In here we have, okay, one second. Here we have ascending vasa recta, here we have descending vasa recta. So again here also fluid is moving up, here the fluid is coming down. So here also opposite direction. So if I combine this both, then this becomes a counter current mechanism. That means this too will have a counter current mechanism, because the fluids will be flowing in opposite direction. This too will have a counter current mechanism. Okay. So basically how many counter current mechanisms? This one, 2, 3 counter current mechanisms. That means counter current when 2 tubes are coming in close proximity with each other, the fluids are flowing in opposite direction. Okay, and why are we doing this? To increase the concentration of the urine, so we can first of all make the osmolarity proper, and with that, students, we also can do reabsorption. We can do reabsorption of fluids. Okay, reabsorption of fluids pannanamna nama vandu oru hypertonic solution create pannanam. Okay, now I'll show you one diagram. Look here, this is the diagram. So here you can see counter current mechanism. Here this is vasa recta, idu vandu loop of Henle. Okay, in the vasa recta 300 osmolarity it is starting, students. Here you can see what is happening. The NaCl is being picked up from here, it is being carried all the way, and it is getting added into the medullary interstitium. Okay, so this is what is happening. From 300 it is increasing up to 1200. Okay, idilume same. What is happening? From the ascending loop, NaCl is getting released out, right? All that NaCl is going to be picked up by the vasa recta here. Adu vandu medullary interstitium la it will get added on. Okay, adukapram here what you see, students, here we are doing water. Water is getting released from the tubules. Okay, and the urea from the collecting duct also is coming to the medullary interstitium, and finally we are going to increase again ingeyum kuda osmolarity from 300. Okay, 300 it is coming up to 1200. So this is how it increases. It does not drastically increase. It goes from 300 to 400, 400 to 600, 600 to 800, 800 to 1000. Adu madri increase agum. So here also remember that the increase is not happening suddenly. It is going from 300, then it is going to 400, 400 to 600, 600 to 800, Apparama, here it is coming up to 1200. Immediately you can see the water molecules are getting released out. Clear? So that is how we are doing this counter current mechanism. I hope it's clear to all of you. Next mechanism on the students, RAS mechanism. Nama vandu already renin how to release we already understood. Yes or no? Yes ma'am, we understood. We did it when? Okay. Okay, see. So we did this. Okay, this is called as your renin. Just juxtaglomerular apparatus. So here what happened? நம்ம GFRல ஒரு ப்ராப்ளம் இருந்துச்சு, that is low NaCl or high NaCl or low GFR, high GFR. And the problem, because of that problem, we activated the smooth cells of efferent arterioles. We formed granular cells, and that granular cells are also called as juxtaglomerular cells. They store and release renin. So on the renin one, the students, I told you it is going to be utilized elsewhere. Adudha RAS mechanism. Okay, ma'am, edukku ma'am ee mechanism, why this mechanism is there? In the mechanism, students, mainly it is going to be there when we have problems in our body. Okay, problems in our body. Like people who do extreme diet, no, sometimes what they do is they cut sugar, they cut salt, everything from their body. Sometimes because of that the salt balance will be different, the sugar balance will be different, ellam vandu different ah irukum. So here the problem is, students, inga vandu GFR romba kammi agum. Okay, GFR will become very less. That is the first problem. When GFR is less, automatically the cells are going to get activated. Blood volume is going to be less. Enagum, blood volume will be less. If blood volume is less, means what? The water also can be less. Okay, and if NaCl is less, okay, imagine when NaCl is less, that means what? BP will be less. Yes ma'am, blood pressure will be less. Idella reduce panna time la students, our body will go through problems, so we have to make sure that nothing happens to us. So adanale RAS mechanism is going to work. Now what is this RAS mechanism full form? Renin, R for renin, angiotensin aldosterone mechanism. Renin angiotensin aldosterone mechanism. Okay, I hope all of you are enjoying, all of you marked your attendance and full josh is there. Okay, renin angiotensin aldosterone system. Ayo, mechanism alla student, system. S thane, S ku system. Okay, system. Idudha nama renin full form. Clear? Now, renin we understood. What is this angiotensin, aldosterone, everything we are going to understand. Now, students, in the glomerulus has already, okay, glomerulus, that is the, I'll write here. Okay, this is the JGA cells. In the JGA cells vandu already renin na release பண்றதுக்கு ready ஆயிடுச்சு. Why? Because the macula densa understood that NaCl is very less, so I have to do this renin release. Okay, so in the cells vandu already it is activated, telling that okay, we have to release renin. So now renin got released. Now how are we going to fix all these issues? In the issues ella fix pannanume. In the issues eppadi fix pandrathu? For that, students, this renin has to come into the bloodstream. So இது தான் bloodstream. Okay, in the bloodstream la vanda already there will be one person who is present. Okay, one person who is present, who is already formed by the liver. Angiotensinogen. இது வந்து inactive form la irukum students. This is already made by the liver. Okay, liver has made it and kept it here, saying that whenever required I will use it. Okay, whenever required I'll use it. Angiotensin has kept it here. Now what will happen? In the renin vanda, from here it is going to come here, and what it is going to do? It is going to activate this angiotensinogen to angiotensin 1. What is it going to activate to? Angiotensin 1. Students, in the angiotensin 1, ma'am, so angiotensinogen could not do anything. So can angiotensin 1 do anything? No, students. Angiotensin 1 also is not completely active. So adanale we have to use one more person. Okay, we have to use one more person, but renin cannot do it. So adanale we are going to take the help of, okay, whom are we going to take the help of? Ma'am, lungs. Okay, We are going to take the help of our lungs and we are going to release. Okay, what are we going to release? Lungs is going to release ACE enzyme, NIACE enzyme, angiotensin converting enzyme. Angiotensin converting enzyme. Angiotensin converting enzyme. In the angiotensin converting enzyme on the students, this is going to convert angiotensin 1 to angiotensin 2. It's going to convert angiotensin 1 to angiotensin 2. Now you will be like, ma'am, at least tell me that this fellow will be able to fix all the issue. Yes, this fellow will be able to fix all the issue. How is he going to fix the issue is the question, right? He is going to fix the issue, students, first by activating. Okay, 1 second. Okay, first he is going to activate whom? He is going to activate our adrenal cortex. He is going to activate whom? Adrenal cortex. Adrenal gland is present just above the kidney. So this fellow will first activate adrenal cortex, and after activating, what will he tell? He will tell, bro, there is an issue, you please fix the issue. Okay, because students, very simple. Renin came, renin converted angiotensinogen to angiotensin 1, which is a very important step. On the step, angiotensin 1 couldn't do anything. So we took ACE, that is angiotensin converting enzyme, from lungs, and it became angiotensin 2. Now this angiotensin 2 will start calling the adrenal cortex, telling that you please release aldosterone. Namakku vandu oru problem vandirukku. What it will do? It will tell the adrenal cortex, you please release aldosterone. Okay, you please release aldosterone. In the aldosterone vandu enna பண்ணுவாங்க students? In the aldosterone vandu, it will allow conditional reabsorption. It will tell DCT, it will tell collecting duct, please do conditional reabsorption. So aldosterone will send signal telling that do conditional reabsorption. Do conditional reabsorption of Na plus. Okay, and water. Why? Because nama problem on the Na is less, water is less. So if they do conditional reabsorption, can the issue be fixed? Yes, ma'am, issue can be fixed. So we are going to fix this two issue, that is this and this got sorted. Now H2O is more means automatically blood volume also will be more. Now we have problem with GFR. Okay, GFR. Now GFR means we have to increase the amount of blood that is coming in. So then again adrenal, this angiotensin will be like, don't worry, I have solution for that also. Angiotensin will be directly involved in doing vasoconstriction of efferent arterioles. Okay, efferent arterioles. Okay, it will do what, students? Vasoconstriction. Vasoconstriction na enna? See, normally nama blood vessel vandu idu maadhiri irukum. Okay, normally blood vessel vandu idu maadhiri irukum. Okay, diluted. Ana nama vandu, let's say I want to decrease the amount of blood that is coming in. Na vandu enna பண்ணுவேன்? Vasoconstriction பண்ணுவேன். இப்படி. Okay, I will constrict, so less blood will come inside. So that is called as vasoconstriction. So from vasodilation, this will become vasoconstriction. So adinala what will happen? The blood that is coming in will be more and the blood that is going out will be less. So GFR will become more and the entire problems got sorted. Is this clear to all of you? Very, very important mechanism. I hope it is clear to all of you. So students, what is happening? Very simple. The problems that we faced was GFR is less, blood volume is less, water is less, NaCl is less, BP is less. Now you don't have to remember the problem, Anna, you should know what is the solution for it. For that, we had told JGA cells that whenever we have problem with our, let's say NaCl or GFR, we have to release renin. So renin release agi, it is going to convert angiotensinogen to angiotensin 1, angiotensin 1 to angiotensin 2. Renin can only convert this inactive form to active form, but that active form cannot do the job. So we have to take ACE from the lungs, okay, we have to take ACE from the lungs, angiotensin converting enzyme, and convert it to angiotensin 2. Angiotensin 2 will do the job, that is, it will activate the adrenal cortex, and the adrenal cortex will release aldosterone, and the aldosterone one, it will do conditional reabsorption of sodium and water. This will increase the sodium and water, automatically increasing the BP, NaCl, H2O and blood volume. Now the second problem that we have was that who is going to increase the GFR? For that we have to do vasoconstriction of the efferent arterioles, because if we do that, then the amount of blood that is coming in, okay, that is going as GFR will be more, and the amount of blood that is leaving will be less. Clear? That will be less. This is what is the entire process. I hope it is super duper clear to all of you. I hope it is easy also. Ouch. Okay. Easy? Okay. If you want to take a screenshot, just take a screenshot. I will move. Okay. Okay. Next one, Lama students. Next mechanism is called as ADH and ANF mechanism. Okay. First, let's understand the full form. This is called as anti-diuretic factor, okay, anti-diuretic factor. In the anti-diuretic factor is also called as what? Vasopressin. Students, in the vasopressin you will study in detail in chemical coordination, but just understand this vasopressin is actually released by the posterior pituitary. Okay, posterior pituitary. Okay, posterior pituitary. In the posterior pituitary, with the help of its neurosecretory cells, will add this vasopressin into the posterior pituitary and tell that you keep quiet, hold this until I tell you, don't release it. So, in the vasopressin or antidiuretic hormone is mainly going to work when there is loss of water. Okay, like in sugar patient and all of this, diuresis can happen. Okay, diuresis. Diuresis means what, students? Loss of water. Excess water is getting lost through the body. So, during this time, loss of water happens extremely, then we are problematic, right? We are in a very bad situation. So, for that, we will release this vasopressin. So, when this happens, vasopressin will be released. In the vasopressin, students, first of all, what it will do? It will exogenously, okay, it will send you signal telling that you are thirsty, drink some water. So, first what we will do, we will try to intake water from outside, okay. That is, activate the thirst mechanism. Activate the thirst mechanism. Second thing what we can do, students, again we can do conditional reabsorption. So, here second thing that we can do is, let's say a person is not drinking water, then we can do is conditional reabsorption of water from DCT, okay, from DCT. This is the two things that we can do. Yes or no? Understood? Understood. Yes, ma'am, understood. Okay. Okay, correct? Done? Conditional reabsorption, okay? Anti-diuretic hormone, okay? H for hormone, okay? Factor I've written. Anti-diuretic hormone. What are we trying to do? We are preventing the risk of loss of water through urine. Clear? So, we have to do water reabsorption. So, water reabsorption, we have to take the help of vasopressin. So, vasopressin will do fluid reabsorption from the kidney. And students, also one more thing you will notice in text, not in NCERT, in other books is this people also do constriction. That is, they will also do constriction of the blood vessel to increase the atrial pressure, to increase the BP. Okay, so they will also do constriction sometime. Okay, blood vessel constriction they do. Why? To increase the atrial pressure. To increase the atrial pressure. Some places you will see this. Okay, in NCERT you will not see much, you will just see conditional reabsorption. Done? I hope it's clear. Renin-angiotensin, there is an image. I can show you that. Okay, here, wait, let's put it here. File is not supported. Okay, students, no problem. I will send you the image. Okay, we will delete this. Yeah, okay, this is the one. So, constriction done? Yes, ma'am. Okay, all of you understood? No doubts, right? Now, we will write ANF also. We have to write ANF as well. Very simple, okay, this is. Wait. Lot of images are there, but the problem is not able to download any of it. There's some problem. Okay, yeah. Next one is, students, ANF, anti-natriuretic factor. Very simple, this is anti-natriuretic okay, ANF, anti-natriuretic factor. Anti-natriuretic factor. Yeah, brain glitch happened. Okay, yeah. So this is the one, students. This one very simple to remember. It works opposite to RAS mechanism. Just remember this. And one more thing that you have to remember, this is actually released from the heart. Okay, anti-natriuretic factor. This is actually the hormone that is released from the heart. This factor is going to work opposite to the RAS mechanism, students. Why, ma'am, opposite to RAS mechanism? Because, students, every time, let's say, we are continuously increasing the GFR. Okay, there should be someone to decrease also, no? Let's say every time the GFR is low, then we RAS mechanism will do what? It will keep increasing the GFR. Yes, ma'am. Imagine RAS mechanism is not in control. It will keep increasing. That time your natriuretic factor will be there, which will do the ulta of RAS mechanism. So, they will ask you question, who is working antagonistic to RAS mechanism? Your answer should be natriuretic factor. It works antagonistic. That means ulta. Fancy word, okay? Antagonistic to RAS mechanism. Very, very important. Very, very important. Can you remember so much? Easy to remember? Yes, okay. Anti-diuretic hormone vasopressin, which will be released from the posterior pituitary. Diuresis should be prevented, that is loss of water through urine. So, for that we will release vasopressin. Vasopressin will also give thirst mechanism. At the same time, it will do conditional reabsorption of water, and it also do constriction to increase the atrial pressure. Okay, increase the atrial pressure. Then we have anti-natriuretic factor, which will do the opposite of RAS mechanism. It is released from the heart. This is what is important for all of you. Okay, understood, ma'am. Now, one more topic that we have, micturition. Okay, one more topic that we have is micturition. Okay, micturition. What is this micturition meaning, students? I think I wrote an A extra. Oh. Board is like, enough. How much are you teaching, lady? Okay, micturition. Micturition means what, students? Basically, expelling of urine. Okay, let's say we want to expel the urine. That process is called as micturition. Expel of urine outside the body. Okay, outside the body. Correct? Yes, ma'am, correct. Okay, expelling the urine. Now, what did I tell you, people? I told you that we are forming students, around how much? We are forming around 180 liters of GFR, glomerular filtrate, right? GF we are forming. This 180 liters of GF are we going to use? No ma'am, we are not going to use. Correct, that much we are not going to use. Now students, what is happening here? Remember, here 180 liters of GF is going to be released. Out of that, only 1 to 1.5 liters will go out as urine. Only 1 to 1.5 liters. That means I told you already, around 99% reabsorption will happen. Reabsorption will happen, and in this 70 to 80% reabsorption is happening in the PCT. Right, in the PCT it is happening. So 99% reabsorption happened. Yes ma'am. Now in this 1 to 1.5 liter students, how much urine will be, how much urea will be there? Around 25 to 30 grams of urea will be there. This also has come as a PYQ of urea will be there. Because we are ureotelic organisms, so obviously urea will come out. Clear? Now I told you that the urinary bladder can store up to 800 ml of urine. 800 ml of urine can be stored in urinary bladder. Okay, in urinary bladder, around 800 ml of urine can be stored. Now students, please remember, how does the whole process happen? Micturition, how does the expulsion of urine happen? I told you, for expulsion of urine to happen, we will need, okay, sorry, okay, for expulsion of urine to happen, we need to send signal, right? CNS, first of all, stretch receptor will tell that I am getting full, like the urinary bladder is full. Okay, urinary bladder will also send signal telling that I am full. Okay, so stretch receptors which are present on the urinary bladder will send the first signal telling that I am full, what to do, because this I told you, brain only will send all the signals. Other than that, no one else can send the signals. They don't know what to do. None of the organs know what to do. So brain will send signal. Okay, now brain will send signal, and the detrusor muscle which is on the urinary bladder will start their contractions. Okay, it will start their contractions, and as the contractions happen, what will happen? The internal sphincter will open, which is not in our control. Okay, urethra will open, internal sphincter will open, then on our knowledge, external sphincter will open, students, and once external sphincter opens, obviously micturition will happen. Now I want you to remember one thing. See, this is the sphincter. Sphincters are contracted, so urine shouldn't come out. They will relax. That is when urine will come out. So that means these people have to relax. Okay, these people have to relax for urine to be expelled out, and this, that is urinary bladder, has to contract. Understood? Okay, understood. One has to contract, another one has to relax. That means urinary bladder has to contract, and when the urine is coming down, your sphincter muscles will be contracted like this, so not to allow any urine to expel out. They will relax, so the urine can come out. So one place contraction, one place relaxation is happening. Understood this? Yes or no? One place contraction and one place relaxation is happening. Understood, ma'am? Okay. Now students, sometimes, okay, I'll do one thing, I'll write this in different color, otherwise you people will think that, okay, everything is part of, okay, wait, we will write this little this side. How much is there? 25 to 30 grams of urea. Okay. Now I told you students, sphincters have to relax. Okay, and the urinary bladder has to contract. Then only this process will happen, because contraction should happen of the urinary bladder. Yes ma'am, contraction of the urinary bladder should happen, and the sphincter muscles which were kept tight like this should start relaxing so as to release the urine out. Correct? Yes ma'am, correct. Okay, this is what is called as micturition. This is what is called as micturition. So this is the entire flow. Sometimes they can also ask you, how the urine is coming. You know now from the medullary, this it's coming. Now students, let me check if I can show here. Okay, if I can show here, I don't think, I doubt if like 3D will be there, but let's try. Okay, I think here it is there, urine formation. Okay, let's see what is there inside this. Okay, it's not visible. See students, here you can see. Can you see? From afferent arterioles, blood is going, and now it will pass through that layers. What is that? Endothelium, epithelium, and the basement membrane. Now the blood is moving. Can you see? Reabsorption will happen in the PCT, and the blood is moving. It is coming where? To the descending limb, where it is going to absorb the water. Okay, and as it goes to the ascending limb, only the electrolytes will be moved out, and now it will go to the DCT. Then it will go to the collecting duct. Okay, see, it is coming out through the collecting duct, right? Now blood is not coming out. Remember that only purification is happening. Okay, only purification is happening. Urine will be coming out. Clear? Urine will be coming out. So for that, blood should go through this process, right? Now can you see, from the efferent arterioles, the blood is moving. Okay, now here you can see reabsorption, secretion will happen. See, this arrow secretion, this is reabsorption. From there, the blood is going all the way, and during this time, filtration is continuously happening. Okay, as they are moving, can you see that? So look here, the movement of substance from the blood within the glomerulus into the capsular space. Tubular reabsorption, the moment of substance from the tubular fluid back into the blood. Secretion, the moment of substance from the blood into the tubular fluid. This is what I told you. Here, filtrate will be moving. Okay, that time here in the arteries, what will be moving? Blood will be moving. Right, blood will be moving, and this is what is vasa recta. Can you see? This is what is vasa recta. Can you see the fluid actually? See the fluid? Okay, fluid formation is happening. Again, see, from here the substance is coming. From here it will go again, it will get into the efferent peritubular capillaries. So this is how it happens. I hope this is clear to all of you. Okay, it's clear. Please remember, it's not blood that is coming out. For us, urine it will be coming out. Okay, urine will be coming out. But from where are we taking all this urea? It has to come from the blood. Right, so blood will be sent back after purification, blood will be sent back, but like this only, same thing like breathing. If I want to add oxygen, I have to bring the blood and add oxygen, like that. If I want to purify the blood, I have to move the blood through the kidney, and the kidney will reabsorb all the unnecessary things from my blood and send the blood back, correct? That is the same thing that is going to happen here, understood? This is how the mechanism happens. You can see blood is right now here, okay, it is here, okay, it will, anywhere movement is happening, I am not able to see. Capsular space, okay. Here. 1 second, students. See here you can see the blood is moving like this, see. Opposite direction, that time fluid is moving like this. Can you see the movement? I don't know, but you can see small grains, okay. Now see again afferent arterioles, okay, afferent arterioles, it's coming, see. It's all, see, next set of blood is going, okay. They'll go now like this, they'll pass all the way through the tubules, okay. It's passing. See, it's going, now it's gone to the ascending lymph, now it will go all the way to the collecting duct, okay. Yes, see, it's moving, okay. We'll see the whole thing, okay. Then you see, students, look here back again. Okay, now from the, see, efferent arterioles, the blood, so it has gone back to the efferent, okay. Now it is coming like this, whatever the blood wants, it will reabsorb, whatever the blood doesn't want, it will give it back, okay. So now the blood is again going, can you see, it's a continuous process, again blood is going, see, it will go all the way and it will come down, see now, see, now again it's moving. Now why? To concentrate it more. It is coming through this way to vasa recta. In vasa recta, concentration will happen, and how much reabsorption we can do, that much reabsorption we can do, and then the blood will again move and enter through the afferent artery. So like this process has to continue. Clear? So this is what is your urine formation. 1 second, students. Okay, I hope this is clear to all of you. Now we have to do, let me see this also once. Okay, once if I see, I feel like okay, I missed out some video and I want to see that also. Load. Loading. It's loading. It's little slow internet it is. 1 second. We will make it big. So here you can see, no? So here we have first, what we have? Juxtaglomerular cells are there. Okay, we have the podocytes. Can you see podocytes? They are arranged in an intricate manner. Can you see full intricate manner they are arranged? See when I am moving it. Okay, podocytes, specialized cells in the kidney. Okay, then students, here what do you see? The parietal cells, their main function in the Bowman's capsule is to act as reservoir and generate the damaged podocytes. Okay, that is not there for you people. But then, see here, this is your efferent arterioles. Okay, this is your efferent arterioles. So on the efferent arterioles, can you see juxtaglomerular cells, and they are present, okay, with the podocytes. Here you can see PCT is there, okay, here collecting duct, here your DCT, okay, and here they go. Here is the loop of Henle, okay, loop of Henle is here. So this is how the whole process is happening. I hope this is understood. Okay, DCT we did. Okay, this is this is DCT, this is parietal cells, efferent juxtaglomerular, you see podocytes, you can see, no, podocytes are present. Okay, done. Now we will go back to our chapter. So this is what has happened. I hope urine formation also is understood. Now students, this is how the urine will be released. Now next is, even though from starting we were telling that, you know, kidney is the one that is doing excretion, we also have some other organs which are also going to help us in excretion. Like example, our lungs remove a large amount of, our lungs are removing large amount of carbon dioxide, approximately 200 ml. So first, who is there? Okay, just write a tabular column and keep, so it becomes easier. Lungs is releasing, it is secreting carbon dioxide, okay, approximately 200 ml. This is important. Wherever numbers are given, questions can come. Okay, 200 ml is giving out per minute, and also significant quantities of water every single day. So lungs are doing what, students? It is doing the job of carbon dioxide and water, okay, removal. Correct? Yes, ma'am. Next, students, liver. Liver being the largest organ, okay, largest gland in the body. It is also doing what? It secretes bile containing substance like bilirubin, biliverdin, cholesterol, degraded steroid hormones, vitamins and drugs. Okay, steroid hormones are the hormones who are made of cholesterol. Cholesterol is a precursor. Okay, so steroid hormones. So here liver is doing the next one. What is liver doing? It is giving out bilirubin. Like this only, write it in your next year NCERT itself and keep it. Okay, bilirubin, biliverdin, cholesterol, okay, steroidal hormones, okay, steroidal hormones as well as some vitamins is being removed by the liver, okay, and drugs. Most of these substances ultimately pass out along with the digestive waste. Now next one is, students, the sweat. Okay, the sweat and the sebaceous glands in the skin can be eliminated certain substances through their secretions. Okay, so through sweat and also oil glands, we will be able to remove some amount of waste materials. Sweat produced by sweat glands is a watery substance that is containing NaCl and small amount of urea, lactic acid, etc. Lactic acid happens mainly when we do workout. Okay, I taught you yesterday, you know, lactate dehydrogenase. Even in our body, lactic acid formation happens. Though the primary function of sweat is to facilitate the cooling effect, with that, they are also giving out some NaCl, some urea, some lactic acid, very small amount. Okay, it also helps in removal of some waste mentioned above. Next one, we have sebaceous gland. They eliminate certain substances like sterols, okay, hydrocarbons and waxes. Okay, waxes can be released through the oils, okay, sebum. Now, this secretion provides a protective oil covering for the skin. Do you know that small amount of nitrogenous waste can be eliminated from saliva as well? So, students, lungs, liver, sweat, sebaceous gland, saliva. This 5 you have to remember and keep, because this 5 is also helping us in removal. Sebum, sweat, saliva. All three are also helping us in removing the excretory waste material. Now, last topic of the day, disorders. I hope by now we have hit more than 150 likes. Okay, don't disappoint me. I want all of you to hit the smash the like button, and also quickly subscribe to the channel. You promised 10K in October. Now October is over. Okay, so irresponsible you people have become. Now students, disorder students, whenever our kidney fails, people tell, no, I can stay with one kidney. Is that true? Yes, it is true. Okay, we can stay with one kidney because filtration happens at such a rate, we can stay with one kidney. But the problem is, let's say two kidneys also failed. Okay, two kidneys also failed, that is leading to kidney failure of both the kidneys. Okay, both the kidneys have become dysfunctional. Then what is the solution for us? Both the kidneys have become dysfunctional. Then our solution is going to be to do artificial kidney. Use artificial kidney, that is called as your dialysis. Students, dialysis is the process where I am removing, okay, removal of nitrogenous waste from blood. Correct? Yes or no? Yes. Okay, I am removing nitrogenous waste from blood. This is what is called as dialysis. Now, understand, dialysis is not the best solution, but it is a temporary solution. That means for some time you can keep doing this. Your permanent solution is only, students, permanent solution is a kidney transplant. Permanent solution to kidney failure. Permanent solution is transplantation of kidney. Okay, graft rejection can happen, but if you are taking the kidney from your let's say relatives, close relatives as donors, then graft rejection chances are very less. All of you know in microbes chapter you will study graft rejection. To prevent graft rejection, we can give immunosuppressants like cyclosporin A injections to control the rejection rate. Okay, so students, what we can do? Dialysis, we also say one more word, hemodialysis. Okay, hemodialysis. What is this hemodialysis? Hemodialysis means we are basically purifying the blood. We are purifying the blood. So I'll give you a simple setup of this, not the exact setup, but the simple setup given in your NCERT. Similarly, only it happens, but more complex it will be. So students, there are going to be tubules. Okay, there are going to be two tubes which are made up of cellophane tapes. Okay, they will be made up of cellophane tapes. So this cellophane tapes will have small tiny pores in them, which will help them to excrete out, okay, like give out the urea. Clear? This is the cellophane tapes. Through the cellophane tapes, what is going to happen? One second, students. I'll draw it a little more spread. Okay. Okay, like this convoluted cellophane tapes will be there. This cellophane tapes, what they will have? They will have tiny, tiny pores. Okay, now what they are going to do mainly, students, please understand. We are going to give the blood, okay. Blood will be given from the patient. Now remember that the blood will, like everything in this dialyzing fluid, there is going to be a fluid inside, right? That is called as the dialyzing fluid. What is it called? Dialyzing fluid. This fluid will have the same composition as the blood, but only one thing is going to be absent, that is going to be urea, because we are trying to give out, right? So if I have urea in it, then it is not going to come out. So the blood is going to pass. This is the patient blood. The patient blood will have all the blood components, okay. Blood plus urea will be there, okay. This is the composition. This will have no urea. Okay, no urea. That is, whatever our body wants, it will not be there. Now what will happen, students? As the blood is coming, the urea or the nitrogenous waste will move from a region of higher concentration to a region of lower concentration, and this way we will be able to purify the blood. Is this understood? Is this understood? So it is a very simple process where we are using again a diffusion type of a setup. Okay, so this is called as, this is the cellophane tape. Tape spelling is what, students? Tape. Okay, this is the cellophane tape. It will have tiny pores. The tiny pores will help in the movement of your urea outside, and the dialyzing fluid will have same composition as the blood except for the urea. Clear? Now students, please remember, as we are taking blood, there is chances that coagulation happens. So here we are going to inject heparin into the patient. Okay, we are going to inject heparin into the patient, which is an anticoagulant. And once we inject the heparin, we know that, okay, coagulation won't happen. By the time we are giving the blood back to the patient, okay, let's say here the patient is there. We are giving the blood back to the patient. We have to give here, anti-heparin. What we have to give here? We have to give anti-heparin. Why, ma'am? Because in our body there is already heparin. So we have to give here anti-heparin so as to prevent any problems from happening. So this is what is called as dialyzing entire setup. So please remember students, permanent solution is going to be transplantation of the kidney. Graft rejection can happen, but if we take it from a close relative, the rejection chances are less. Hemodialysis, basically we are removing the nitrogenous waste from the blood, which is a temporary solution. In this dialyzing thing, you will know that there is dialyzing fluid. Okay, there is dialyzing fluid. It will have the same composition of the blood of the patient. Okay, same composition, same. And students, a very, very painful, at the same time very financially draining process. Okay, same composition as blood except for NW, nitrogenous waste, or urea also you can write. Okay, NW. Clear? Now students, please understand here, it's a very, very painful process and it is financially very draining because it's a very, very expensive process. Since it's a very expensive process, it's difficult for people to afford it. Everyone cannot afford it. Maybe 1 sitting, 2 sitting, 3 sitting they can do, but after that it becomes difficult. And for some people it might be told them in 2 months, in 3 months, 6 months. It varies people to people. Okay. Now when a new kidney, let's say I want to get, I have got a kidney, then where do I attach it, ma'am? You can attach it directly to the ureter. You don't have to remove the already existing kidney. Okay, this is one thing I want to tell you. Most of the time students get doubt, ma'am, kidney transplantation means that both the kidneys will be removed. Students, both the kidneys will be removed if there is an infection. Okay, if there is an infection and it will get spread, then it will be removed. Otherwise, in most of the scenarios, they will just connect it directly in the lower abdomen. Okay, here the newly received kidney will be connected like this. Okay, like this it will be connected. So always remember, we are not going to remove the kidney until the kidney has some infection, something that is very problematic. It will affect the entire body. That time only we will remove it. Okay, clear? Now we are going to the last topic, disorders. What is the disorders? Here you have to study 3 disorders. First one is called as renal calculi. So students, renal calculi also called as kidney stone. Very casually we all know kidney stone. It can come to any one of us if we do not drink enough water. Okay, kidney stone if we do not drink any water. Why? Because here what will happen? Stone or insoluble mass of crystallized salts like oxalates are formed within the kidney. Most of the time for this kidney stone, people will recommend to drink more water. Surgery or operation will not be told immediately, but in severe scenarios, extreme abdominal pain and all, they can tell you that okay, you have to go for a surgery. Okay, or a tiny surgery. Okay, so renal calculi, kidney stone, where stone or invisible mass of crystallized salts are present, mostly oxalates, which are formed within the kidney. Next one is called as glomerulonephritis. Glomerulonephritis, that is inflammation of glomeruli of the kidney. Okay, inflammation of glomeruli of the kidney. That means the glomeruli will get inflated. Inflated means what, students? It will become red and swelling will happen. Okay, swelling will happen. Inflammation of glomeruli of the kidney. Presence of glucose, that is glycosuria and ketone bodies, ketonuria. So presence of glucose in your urine, it shows that there is more glucose. Okay, more glucose. And when there is ketone bodies present, it is called as ketonuria. Okay, and the urine are indicative of diabetes mellitus. Diabetes mellitus means what, students? A condition where your body has more glucose. Okay, glucose is more and insulin in the body is not able to break down all of this. Okay, glucose is more and insulin in the body is not able to break down enough. So you will have to take injections from outside. That means you have to take insulin injections from outside so as to prevent this whole problem. Okay, to prevent this whole problem. Clear? This topic is done and dusted. I hope all of you are going to do some main things that I've asked you to make sure that you check the playlist. Let me just show you because most of you are confused. And students, notes will be updated on the sheet. Okay, I'll give you a sheet. Don't worry. Wait, let me just open it here. Huh. So I'm bringing it here for all of you. So all of you know this is the channel. Yes, ma'am, this is the channel we know. So in this channel, now where do I find all the videos, ma'am? Go here. Can you see playlist? Click on playlist. Here, can you see? Class 11 Biology, NEET 2026 Biology one shot. Class 11, Class 12 separately also we have put. One shots also we have put. So when you go, where did it go? So when you go here, just click on this. Okay, you will see respiration is here. Can you see here? Respiration, reproduction, animal kingdom, photosynthesis, human reproduction, breathing and exchange, molecular basis of inheritance. Now excretion also will be added into this list. So we are completing the chapters as soon as possible. Then, okay, one second. Now if you go here, students, some PYQ practice sessions also have happened. And here if you click on tips and tricks, okay, here you will be able to see mock test strategy, only for girls, 0 to aims, are one shots helpful, average to topper, one challenge I had given, how to prepare for NEET using YouTube, series about NEET class 12 timetable is there, NEET difficulty level, okay, all this is here, okay, all this is here. Make sure, see 6 months ultimate plan, latest NCERT syllabus, how to read NCERT, 650 without coaching possible, books that you are supposed to refer. Like this, lot of videos are on the channel. So please do check it out and don't wait for, one good moment to come to, do something. Just check it out right now itself. So you're not going to waste any time. Okay. And students, yes, let's subscribe to the channel as well, because this is the channel you can see solely my face only you can see on the channel. So there's a lot of work going into this channel for all the Tamil Nadu students and lot other Andhra students, Kerala students, all of you have subscribed, I know. But still, I want all of you to subscribe. Let's make it 10K. We are at 9.19K. So I think 10K is possible if all of you show your love, okay, and all of you show me that, yes, ma'am, we really want to do this, okay. So thank you so much, students. See you all in the next class. Till then, take care. Bye-bye. Okay, have fun and also let me know in the comments how the class was. We will do PYQ practice of this. Don't worry. And tomorrow I have some surprise for you. It's coming soon. You will get to know what is the surprise and you're going to be super duper happy. So watch this one shot till the end because the surprise will be surprised. Okay, so bye-bye. Take care. See you all in the next class. Bye, Pasangla. Give lot of love in the comment section. Let me know how the class was. Okay, bye-bye.